Now, let us proceed with the transmembrane proteins and membrane transport. Okay, so before guys, pag pinag-uusapan yung transportation, okay, we are always referring to active transportation, passive transportation. Kaso guys, the main problem is, hindi lang pala ganun kasimple yun, di ba? Napakarami palang klase ng passive and even active diffusion. Okay? So today, iisa-isahin po natin yun. Okay? Anong ibig sabihin ng passive? Anong ibig sabihin ng active? And sino-sino po yung mga passive and active transportation? So first, we have here the passive processes. Okay? Now, in passive processes, or yung tinatawag nating diffusion or passive transportation, this is the movement. Okay? This is the movement of substance. Okay? Movement of substances down a concentration gradient due to, due to the kinetic energy of the substance. No expenditure of cellular energy. So, walang energy kailangan dito. Continues until equilibrium is reached. Okay? So, guys, again, ang sabi, ang passive transportation or passive processes or diffusion, it is the movement of substances down a concentration gradient. Okay? Until what? Until it reaches its equilibrium. Kailangan mag-balance. Kaya nga lagi natin sinasabi, hindi pwedeng mawala yung homeostasis, most especially sa cell. Diba? Kasi kapag nasira yung homeostasis ng lahat ng bagay, okay, sa mismong cell, dyan napapasok yung mga sakit. Okay? And of course, pag sinabing passive, okay, or diffusion, meron tayong tatlong pag-uusapan dyan. Okay, we have three types of passive processes or diffusion. The simple, facilitated, and the osmosis. Okay, so alam mo na kagad, okay, simple, facilitated, and then osmosis. So simple passive processes or simple diffusion, facilitated diffusion, or facilitated passive processes, then osmosis. Okay, so kabisado na. Next, simple, facilitated, osmosis. Now, in simple diffusion, sabi natin ha, walang energy na kasama, no expenditure of cellular energy. Unassisted net movement of small nonpolar substances down their concentration gradient across a selectively permeable membrane. Eh guys, obviously naman, alam natin yung selectively permeable membrane. We are referring to your cell membrane. Diba? We are referring to your cell membrane. And do not forget, when it comes to diffusion, it is from higher concentration to lower concentration. So, sulat natin, no? Higher concentration to a higher concentration to lower concentration lagi. Okay? Kasi syempre, no, marami na nga, dadagdagan mo pa ba? Di ba hindi na? Until it reach its equilibrium. Okay, yun po ang ibig sabihin niya. Now, I want you to remember, pag sinabi pong simple diffusion, this is very much applicable for lipid soluble substances. Okay? For lipid soluble substances. Okay? So, an example for simple diffusion is the exchange of oxygen and carbon dioxide between blood and body tissue. Okay? So, yung po palang gas exchange sa blood is a simple diffusion. Okay? So, madali lang, no? Ang gas exchange, okay, is an example of a simple diffusion transportation. So do not forget. Next. So tapos na tayo kay simple, di ba? So next is the facilitated. It is the movement of ions and small polar molecules down their concentration gradient. So higher to lower concentration pa rin. Assisted across a selectively permeable membrane by a transport system. Now, anong pinag-uusapan dito? We are referring to the movement of ions and small polar molecules. Okay? So, anong ibig sabihin yan? Okay? I want you to remember, kapag po sinabing facilitated diffusion, okay, meron po tayong mga special structures na kailangan to facilitate the transportation of several ions or molecules. Ano yun, ma'am? Meron po tayong tinatawag na mga channels. Okay? So, yung channels na yan, kailangan po natin, di ba? Bakit? Okay? So, kailangan po natin yung mga channels na yan to facilitate the transportation. Another one is carrier. So, ang facilitated diffusion, okay, hindi lang siya movement of ions and small, ganyan, etc. Guys, hindi. May dalawang klase ng facilitated diffusion. Okay, dalawang klase. Imagine mo, no? Ang pinag-uusapan lang natin dito is the passive, di ba? Passive process or passive transportation. And yet, okay, ngayon, alam mo na mayroon pang tinatawag na osmosis, facilitated, and simple. And under the facilitated diffusion, we have the channel facilitated diffusion, okay, or the carrier facilitated diffusion, or yung tinatawag nating mga channel mediated, carrier mediated. Okay? So again, we have two types of facilitated diffusion, the channel mediated and the carrier mediated. Okay? So, ma'am, ano po ibig sabihin? Okay? Let's start with the channel mediated. Guys, this one is a facilitated diffusion. Dalawang klase ang facilitated diffusion para po hindi tayo malito, paulit-ulit. Okay? So, we have here the channel mediated. At sino daw yung isa? The carrier mediated. Anong ibig sabihin ng channel mediated? Movement of ion down its concentration gradient through a protein channel. So, anong ibig sabihin? Kailangan mo daw ng channel. At ano yung channel na yun? Gawa sa protein. Okay? So, ulitin ko ha. Pag sinabing channel mediated, okay, we have these proteins, okay, that serves as a channel. So an example of channel mediated is sodium moves through sodium channel into the cell. Okay? So in short guys, yung pagpasok, ayan na, yung pagpasok po ng sodium papunta sa loob ng cell, guys, that is a channel mediated diffusion. Okay? Or channel mediated facilitated diffusion. Di ba ang haba? Okay? Okay. Next, we have here the carrier mediated. So sabi nga natin, in facilitated, we have two types of facilitated diffusion. The channel mediated and the carrier mediated. Now, in carrier mediated, movement of small polar molecule down its concentration gradient by a carrier protein. Okay? So parang paulit-ulit lang. Okay, higher to lower concentration. Okay? Kaso guys, anong difference? Ito, may carrier protein tayo na tinatawag. Okay? May taga-assist. Okay? May taga-assist na protein. Okay? Para lang makapasok yung isang ion na yun papunta sa loob ng cell. Okay? So an example is the transportation of glucose into cells by glucose carrier. Okay? Although marami pong klase ng mga glucose carriers, di ba? Okay? But remember, the transportation of glucose into the cell that is carrier mediated. Okay? Yung sodium papunta sa loob ng cell, that is a channel mediated. Okay? Now, do not forget, balik ako dito. The basis for channel mediated, okay, the basis for channel mediated is the size and charge of the particles. Okay, size and charge. Okay, but how about for the carrier mediated? Okay, o balik tayo doon. Sa carrier mediated, so sabi natin kanina, it requires a carrier protein. Okay, guys, ang basis dito is the shape and the charge of the particle. So there is a difference. Bakit? Yung channel mediated, the basis is the size plus the charge. And then for carrier mediated, the basis for the transportation is the shape of the particle and the charge. Okay? So, kita po yung difference naman, di ba? Next. Okay. So, dito po, no, kita po natin si simple diffusion and the facilitated diffusion. So, as you can see, in simple diffusion, okay, walang kahit anong taga-assist. But in facilitated diffusion, dito sa picture na to, we have this protein channel. So, in short, this is a channel mediated, diba? Channel mediated facilitated diffusion. Okay? So, yun po yung difference nila. Pag sinabing simple diffusion, walang special structure na kailangan. Okay? Para makapasok sa loob ng cell. Okay? So, that is the difference. Next. So, dito po, mga kapatid, no, it shows here, again, the simple diffusion, the channel mediated, the carrier mediated. Okay? So, ano pong nakikita po natin, di ba? Pag sinabi nating simple diffusion, ulitin natin, hindi mo kailangan ng kahit anong taga-assist. Okay? But, for the channel mediated, okay, the channel mediated, ano daw ang basis natin? Your basis is what? Your basis is the size. Okay? As well as what? As well as the charge. In carrier mediated, paulit-ulit po tayo, the basis is the shape, okay, and the charge. So, ibig sabihin, dun sa channel mediated na yan, ito po yung protein po natin. Okay? Nagawa daw saan? Sa protein. Okay? Which is somewhat similar to a carrier-mediated facilitated diffusion. However, okay, yung pinaka-carrier nila, okay, ay very specific pagdating sa shape. Okay? So kung pabilog yan, hindi yan papasok dito. Kasi nakikilala niya lang hexagonal. Diba? Okay? So parang ganun lang po yan. So that's the difference. Okay? So I hope guys, medyo naliliwanagan tayo, no? So again, nasa passive pa lang po tayo or yung tinatawag nating diffusion. And remember, we have different types of passive diffusion. The simple, okay, the facilitated, and the osmosis. Okay, we are done with simple. And then facilitated, we have two types of facilitated diffusion, the channel mediated and the carrier mediated. Okay. So last sa passive, we have here the osmosis. So ano ibig sabihin, guys? Take note. Diffusion of water across a selectively permeable membrane. Direction is determined by relative solute concentration. Ayan. Continues until equilibrium is reached. Okay? So, ma'am, anong ibig sabihin yan? What is the example? Okay? Nang merong osmosis daw. Okay? Or gumagamit ng osmosis as the means of transportation of ions. Guys, basically water. Diba? So water daw po, osmosis. Eh ma'am, bakit po ang lagi pong sinasabi ninyo sa amin before or laging sinasabi po ng ibang professors, passive daw po ang transportation ng water. Bakit? May mali ba? Di ba wala? Dahil ang osmosis ay passive. Di ba? Ganun lang po yun mga kapatid. Okay? So take note. Okay. So here it says osmosis, that is the water movement across the membrane. So makikita po natin, di ba? Meron po tayong mga unassisted. Okay, unassisted. Ayan unassisted diffusion. Okay, ibig sabihin unassisted transportation, and we have also a facilitated one. Okay? So, ito po guys, yan po yung tinatawag nating osmosis. Okay? Kasi water pinag-uusapan natin eh. Okay? Kaso dito, merong special structure, okay, which facilitates what? Which facilitates the movement of the water across the membrane. And sino daw yon? The aquaporin. Okay? So, ito po, mga kapatid, itong aquaporin na to, it is a protein, okay, that facilitates the movement of water. Kaya nga aqua water. Okay? So, ibig sabihin, kahit saan dyan, pwede si water sumuot, diba? Pwede siyang sumuot dyan. Pwede rin dito sa area na to. That is osmosis. Okay? So, wag po kayong magtataka pag tinanong ano daw yung transportation ng water, guys. Definitely, it is passive transportation. Yun lang, hindi siya naka-specify. Okay? Pero kung specific ang hinihingi sa inyo, it should be osmosis. Okay? So, next SEM, I guess, marirealize ninyo, ito pala yung sinasabi ni Ma'am Ces. Okay, kasi sa ibang references, sa ibang textbook, hindi po ginagamit yung osmosis. Ang ginagamit is passive. Okay? So, wag na kayo makipagtalo. Totoo naman yun. Diba? Tama yun. Hindi ko sinabing mali. Tama na passive. Okay? Pero uulitin ko, ito ay more specific. Okay? So, next. Now, let us proceed to the active processes. Okay? Now, ano pa ibig sabihin ng active process na yan? Or active processes? That is the movement of substances. Okay? Requires expenditure of cellular energy. So, kanina sa passive, hindi mo kailangan ng energy. But in active processes, it requires cellular energy. Now, we have two types of active processes, okay, or active transportation. The active transport, ayun okay, kaya nga iniba ko yung dito, active process, okay, dalawang klase, active transport and vesicular transport, para hindi na po kayo malito. Okay? So again, active transportation, vesicular transportation. Okay, those are the two types of active processes. Now, we have here the active transportation. Ayan. So marami pong klase ng active transportation ha. Baka po kasi malito po tayo. It refers to transportation of ions or small molecules across the membrane against a concentration gradient by transmembrane protein pumps. Okay? So, guys, kung titignan po natin, halos parehas lang naman yung sa passive and sa active transportation. Okay? But the only difference is what? The expenditure of the cellular energy. Pero halos parehas lang sila kasi yung goal naman nila is to transport different molecules or ions. Diba? Now, maraming klase ng active transportation. Guys, imaginein mo kanina sa passive or diffusion. Diba? We have simple, facilitated, osmosis. Sa facilitated, meron ka pang channel mediated, meron ka pang carrier mediated. Ngayon sa active process, okay, sa active process, meron kang tinatawag na active transportation, di ba? And then vesicular. Okay? So active and vesicular transportation. Sa active transportation, hindi pa doon nagtatapos yun. Bakit? Meron pang primary, secondary, symport, antiport. Di ba? Toxic. Okay? Mambalik na lang po tayo sa passive and active para mas madali. Hindi, kailangan kasi natin ito. Okay? Para maintindihan po natin. Okay? So, primary, secondary, symport, antiport. Now, ito guys, definitely kakabisaduhin ninyo ito. Okay? Pero, meron akong gustong ipaintindi sa inyo dito. Okay? What is the difference between the primary and the secondary symport, antiport? Okay? Eh ma'am, nakasulat naman na dyan eh. O sige, given. Basahin ko sa inyo para mag-gets natin ha. Although alam ko naman matatalino kayo. Primary. Okay? Movement of substance up its concentration gradient powered directly by ATP. So obvious yun. An example is calcium pumps transport calcium out of the cell. Sodium potassium pumps move sodium out of cell and potassium into the cell. Okay? Secondary, movement of substance up its concentration gradient, okay, is powered by harnessing the movement of a second substance, okay, sodium down its concentration gradient. Wala ko naintindihan, ma'am. Symport, movement of substance up its concentration gradient in the same direction as sodium. Antiport, movement of substance up its concentration gradient in the opposite direction from sodium. So, anong ibig sabihin yan? Okay? Pwede po bang, dun muna tayo sa primary and secondary. Diba? Primary, secondary lang tayo. Sa primary, movement of substance up its concentration gradient powered directly by ATP. So, very simple. Ang kailangan mo lang is ATP. Okay? Ang kailangan mo lang dito is ATP para lang magkaroon ng transportation in and out. Diba? Yung sa secondary, sabi dito, movement of substance at its concentration gradient is powered by harnessing the movement of a second substance. Now, what is that second substance? Binigyan ka ng example, sodium, down its concentration gradient. Okay? Eh, ma'am, wala ka namang example dyan. Guys, bigyan ko kayo ng isang magandang example. Meron po tayong tinatawag na NIS. Okay? NIS. What is NIS? O, may nakakaalala po ba sa mga nag nag-CC na dyan? Okay? As in CC na clean chem ha, hindi nag-CC na nag-clean na nag-third year, 'di ba? So NIS, what is NIS? Guys, ito po yung tinatawag kasi nating sodium kasi natrium, 'di ba? N I for iodide. Okay, sodium iodide import. Okay, ito yung pinakamadaling ibigay ko sa inyo ng example dito. Okay, sodium iodide import. Ma'am, anong connect? Ma'am, teka, nako-confuse ako. Ang sabi mo, ma'am, secondary, ma'am, iba pa po yung import. Okay, iba pa po yung import. Yung sodium iodide, okay, is secondary. But it is also a import. Anong ibig sabihin ng import? Movement of substance at its concentration gradient in the same direction as sodium. Same direction. So, kasi guys, itong NIS na yan, para maintindihan po natin. Okay, drawing tayo dito ng konti. Okay, ayan. Drawing lang ako. Okay. Guys, katulad dito, let's say this is your thyroid follicle. Ito kasi yung isa sa pinakamadaling example. Okay, thyroid follicle. Andito si blood vessel. Inside your blood vessels, nandun yung mga different nutrients, di ba? Andun si sodium, okay? Andun si sodium, andun si iodide. Okay? Andito sila sa blood vessel. And marami pang iba na nasa blood vessels. Okay? So, anong sinabi sa secondary? Hindi lang enough. Ayan ha, hindi lang enough na may ATP ka. Okay? Hindi lang enough. Bakit? Kasi kailangan mo ng isa pang substance na sumama. Ayan ha, na sumama doon sa isa. So in this case, okay, sino ang sasamahan? Si iodide. So para ma-pump paloob si iodide, kailangan mo ng sodium para makapasok sila. Which is, same principle din po ng import. Anong sabi? Same direction as sodium. Yun ang ibig sabihin niya. Okay? O, yun guys ha, para lang po maliwanag po sa atin. Okay? Secondary. Okay? Kaya nga po, dapat alam mo lang kung primary or secondary. And then, tignan mo, same direction ba ng sodium or opposite direction? Yun. Pero ang totoo, primary, secondary ka lang naman. Okay? Ma'am, parang synthesis ng thyroid hormone yung sa secondary ko. Yes, pero hindi yung kumpletong ano ha, hindi yung kumpletong synthesis ng thyroid hormone. Yung pagpasok pa lang ng iodide kasi guys, sa thyroid hormones kasi ano sila eh, kailangan nila ng iodine. Ayun na, iodine, not iodide. Okay, mahabang proseso pa yan. Pero yung initial, kailangan ng secondary transportation. Okay? Or yun yung tinatawag nating NIS na yan. Sodium iodide synthol. Okay? Para makabuo ka nga nung mga thyroid hormones. Pero initial lang. Okay? Hindi pa kabuuan niya. Mahabang discussion. So may idea ka na about import. Diba? Nagets mo na, ay, lang pala yun. Dali-dali naman. Okay? Then next, antiport. Guys, anti nga eh. Nag-o-oppose ka. Kanino ka nag-o-oppose? Kay sodium. Okay? Okay, kay sodium. So, napakadali, di ba? Alam mo na kagad, import, antiport. Okay, ang dali niyan, ma'am. Alam ko na yan, ma'am. Okay. Lipat ka na. Okay, next. So, katulad nito. Okay. Sa primary, sabi nga natin, ito lang titignan mo. Primary, secondary. Alamin mo lang dapat kung primary or secondary. Okay? So, dun sa primary active transportation, guys, take note, di ba? Kapag kailangan mo ng ATP, ayan, that is primary. Okay? That is primary. Ma'am, ano nga po ulit kapag secondary, guys? Take note, meron kang second substance. Sino yung second substance mo? That is the sodium. Ngayon, tignan mo kung opposite direction ba siya or hindi. Okay? Katulad dito, opposite direction siya, yung potassium and sodium. So, that is antiport. Dito, okay, sodium and glucose, papasok. Same port, kasi same direction. Diba? Madali lang siya. Okay? Medyo nakaka-confuse lang kapag binasa mo, pero ang totoo, ganun lang palang kasimple yun. Basic. Diba? So, next, vesicular transportation. Okay? Or vesicular transport. So, familiar na po tayo, no? Sa active process. Okay? We have this active transportation. Okay? And then the vesicular transportation. So we are done with the active transportation such as primary, secondary, and we were able to differentiate symport from antiport. Okay, so familiar na. Next, vesicular transport. Ano ba yung vesicular transport na yan? In vesicular transport, vesicles, okay, or vesicle formed or lost as material is brought into a cell or released from a cell. Okay, kaya nga meron po tayong tinatawag na exocytosis or endocytosis. Okay, so obviously, if that is exo, palabas ng cell, while for endocytosis, paloob into a cell or released from a cell. Ito yung release, ito yung into a cell. Okay? And then in endocytosis, meron tayong tinatawag na phagocytosis, pinocytosis, and the receptor-mediated endocytosis. Guys, imaginein mo, transportation lang to, no? Pinahaba-haba, active-passive na lang sana pinag-uusapan natin yan. Okay? So, again, in endocytosis, we have the phagocytosis, the pinocytosis, and the receptor-mediated endocytosis. Okay. Now, exocytosis. Umpisahan natin kay exo. That is the bulk movement of substance out of the cell by fusion, okay, of secretory vesicles with the plasma membrane. Guys, remember, release kapag exo. Galing sa loob ng cell, palabas. Okay? So, madali lang. So, the example of exocytosis is the release of neurotransmitter by nerve cell. Last time, nagbigay tayo ng example ng neurotransmitter. Natatandaan niyo po ba kung sino yon? Meron po bang nakakaalala kung sino yon? Baka wala na, ha? Okay. So, mukhang wala. Acetylcholine. Ayan, acetylcholine. Good. Okay, acetylcholine. Okay, acetylcholine. So, so very good. Okay lang mali. Okay, okay lang yun. Natututo tayo sa pagkakamali natin. No? So, okay. So, next. Endocytosis. Oh guys, itong mga susunod na to, puro paloob na to, ha? Okay, paloob na to. Okay, endocytosis. Bulk movement of substance into the cell by vesicle forming at the plasma membrane. Okay, so ang dali, no? Anong keyword natin dito? Movement of substance sa exocytosis out of the cell. Pero endo into the cell. Napakasimple, ma'am. Ano ba naman, huwag na ituro sa amin yan. O sige, phagocytosis, pinocytosis, and receptor-mediated endocytosis. Okay, what is phagocytosis? Phagocytosis means that is the engulfing. Di ba? So, ma'am, ano ibig sabihin? Vesicles are formed as particulate materials external to the cell are engulfed, okay, by pseudopodia. Guys, ano ibig sabihin ng pseudopodia? Di ba, nagpara kayo? Ano ibig sabihin? False fit. Di ba? False fit yan. Hindi totoong mga paa yan. Okay? So, kaya nga meron po tayong tinatawag na mga phagocytes. Di ba? Okay? And yung mga phagocytes na yun, example is neutro, di ba? Neutrophil. Ayan, neutrophil. And monocytes. Ayan na lang, monocyte na lang. Neutrophil, neutro. Ayan, neutrophil and monocyte. Okay? Sila yung mga phagocytes na tinatawag. Di ba? Guys, kapag sila active, nagkakaroon sila ng mga pseudopods. Most especially yung mga monocytes natin, kaya nga very irregular ang shape nila eh. Kaya nga pag po tayo nag-aral na ng blood, sa totoo lang, sabi niyo, ma'am, kabisado ko. Alam niyo, kahit kabisado ninyo yung mga cells na yan, you can really define the cytoplasm, the size, di ba? The number of nucleoli or nucleolus or nucleus or nuclei, kabisado mo, pero hindi mo siya talaga ma-identify kapag picture na ang pinag-usapan. Okay, kunwari, identify this cell. Ma'am, sabi po kasi gray cytoplasm eh. Grayish cytoplasm. Ma'am, sabi po kasi, ma'am, ganito. Guys, hindi na siya totoo. Bakit? Kasi it really moves, especially kapag active. Kaya nga may pseudopods. Diba? Pseudopodia. Okay? So, the example is engulfing a bacterium. Ayan, nabanggit ko na. Pinocytosis. So, ito po yung pag-inom, di ba? Kaya nga, interstitial fluid is taken up by the cell. Yung phagocytosis is eating. Dito sa pinocytosis is drinking. Okay? So, example is formation of small vesicles in capillary wall to move substances. Okay? And the last one is the receptor-mediated endocytosis. Okay? Receptor-mediated endocytosis. So, paano po yan, ma'am? Plasma membrane receptors first bind specific substances. Okay? Receptor and bound substance then taken up by the cell. Okay? So, ano ibig sabihin yan? Okay? Plasma membrane receptors first bind to specific substance. So guys, kasi sa isang cell membrane, normally, meron silang mga receptors. Okay? Meron yung mga receptors. And yung receptors na yan, kapag may nag-bind na dyan, i-engulf na niya yan para makapasok sa loob ng cell. Yun po yung ibig sabihin ng receptor-mediated endocytosis. Okay? So, tandaan po natin yan. Medyo familiar na tayo, di ba? We have the exo and the endocytosis. In endocytosis, meron po tayong phagocytosis, pinocytosis, and the receptor-mediated endocytosis. Okay? Okay. So, dito po, no, pinapakita na meron tayong endocytosis and the exocytosis. Of course, guys, pag sinabi natin endocytosis is paloob, di ba? Kaya nga taken up by the cell or into the cell. Okay? So, food particle taken in by endocytosis. So, food particle. So, ayan. Nagkakaroon tayo ng vesicle inside. Diba? Ito na yun. Nagkaroon tayo ng vesicle. Okay? Ito na yung mismong vesicle, mga kapatid. Kaya nga vesicular transportation. Okay? And then, ang example po dito is the lysosome. And remember that your lysosome is the suicide bag of the cell. So in short, kung ito po ay, let's say, bacteria or bacterium, so anong gagawin po ni lysosome sa bacteria or sa bacterium? Of course, kakainin niya yan, di ba? Papatayin niya yan. Okay? Ida-digest niya yan. So dahil na-digest na niya, guys, hindi na kailangang maipon dito sa loob. Anong mangyayari? Ilalabas na niya yan. Okay? Bakit? Wala nang silbi yan. Diba? So that is exocytosis. Okay? So, yun po yun mga kapatid. Naiintindihan na po natin ngayon, okay, yung endo and exo. Okay. So in endocytosis, taken by the cell, di ba? Taken up by the cell. Okay? Meron tayong phagocytosis, pinocytosis, and the receptor-mediated endocytosis. Okay? So kung mapapansin po natin, in phagocytosis, we have here the pseudopods, yung false teeth, di ba? Para makain po yung ating antigen or any solid particles or kung ano man yan. Diba? And then it will form a vacuole. Okay? And then, pag pinocytosis, guys, of course, we are referring to fluid. Kaya nga, inom drinking. Okay? Wala nang pseudopods. Okay? And it will form a vesicle. Then, receptor-mediated endocytosis. Guys, anong meron daw dito? We have here the receptors. Okay? So, yun po yung tatandaan lang po natin. Para at least, kung picture man po ang ibigay ko sa inyo sa quiz or activity, you will be able to identify, okay, kung anong process yun, kung anong klaseng transportation yun. Okay? Next. Now, let us proceed with the stages of phagocytosis. Okay? Siyempre, guys, itong stages of phagocytosis na to, kailangan sunod-sunod. Hindi po pwedeng, okay, chemotaxis, sunod, digestion, sunod, kung ano man, di ba? Kumbaga, nasira na yung arrangement na to, yung stages na to. Remember, stages nga pagkakasunod-sunod. Okay? So, we have the first one, recognition, chemotaxis, ingestion or engulfment, and then the digestion. Okay? So, ma'am, ulit po. Recognition, chemotaxis, ingestion or engulfment, and then the digestion. So, ma'am, anong ibig sabihin yan? Phagocytosis to, ha? Remember, phagocytosis is a form of endocytosis. Okay? So, remember, kanina meron tayong mga tinatawag na phagocytes. Okay? Now, ma'am, anong meron dyan? Example, meron ka po dito antigen. Okay? And then, andito si WBC. Okay? First, the WBC will recognize the antigen. Di ba gano'n naman talaga yun? Kunwari, ikaw, okay? Kunwari si Ivan. Ayan, si Ivan. Okay? Nakita si Dantes. Okay? Ito si Dantes. Oh. First, na-recognize mo siya. Di ba? From afar, na-recognize mo siya. Okay? So, anong gagawin ni Ivan or ni Dantes? Lalapit. Di ba? So, that is chemotaxis. Okay? O hanggang doon lang, hindi na po yung engulfment and then digestion. Hindi na po yan. Okay? Ang pinapakita ko lang po, baka kasi mamaya sabihin, ay i-ingest ni Ivan si Dantes. Hindi po. Okay? Kumbaga pinapakita ko lang sa inyo na kailangan mauna yung recognition before yung chemotaxis. Kasi yung chemotaxis is the movement of the cell towards the foreign body. Diba? Yun ang chemotaxis eh. Okay? And then after that, di ba, magkakaroon na ng ingestion or engulfment and then digestion. So balik tayo kay antigen and kay phagocyte. Okay? Na-recognize ni phagocyte si antigen, therefore lalapit siya, of course, because of their pseudopods. Okay? Lalapit sila dito, okay, sa antigen, and then it will engulf. Okay? And then it will digest. And lahat po ng mga na-digest nila for excretion, it will be excreted out from the cell through exocytosis. Yun ang ibig sabihin po nito. Okay? So do not forget the different stages of phagocytosis. Okay. So, another example, ito ulit, di ba? We have here the pseudopods, okay? The particle, i-ingest, okay? Ayan. Nilapitan, di ba? Nilapitan because of their pseudopods. Then, it will form a phagosome, okay? And then, phagolysosome, ibig sabihin, may kakain na lysosome, di ba? And then, ida-digest po ni lysosome, di ba? Pagka na-digest na, magkakaroon tayo ng residual body and then, i-excrete. Okay? Yun po yung sa phagocytosis. Okay? Magagamit niyo yan. So next, so we are done with the cell membrane. Imagine mo, no, intro lang pala yun. No? Cell membrane pa lang po tayo mga kapatid. Now, punta na tayo sa cytoplasm. Okay? Guys, do not forget, pag sinabing cytoplasm, okay, it is the gel-like material located inside the cell, which consists of a fluid matrix in which the organelles, inclusion, and nucleus are suspended. Ano ibig sabihin? Okay? Guys, hindi po tama na tinatawag mo na cytosol ang cytoplasm. Okay? Inuulit ko po, hindi po tama yun. Okay? Iba ang cytoplasm sa cytosol. Okay? Yung mismong cytosol, okay? Yung mismong cytosol, ito po yung mismong fluid. Okay? Fluid. Okay? Fluid lang. Okay? But remember, the viscosity of the cytoplasm depends on what is present inside the cytoplasm. Kasi may kinalaman po yung mga organelles, di ba? Sa viscosity ng mismong cytoplasm natin. Okay? So, guys, tatandaan po natin. Kaya pag po tinanong ko kayo, which part, okay, of the cell consists of a fluid matrix, guys, hindi, ma'am, pwede po cytoplasm / cytosol. No. Okay, hindi po. So, kinlaro ko lang po yun. Next. So, inside the cytoplasm, of course, the main component of the cytoplasm is water. It is about 75%. Okay? And then, meron din po siyang salts. Okay? Huwag kayong magtaka dyan dahil meron kang sodium and chloride sa katawan mo. Okay? It also contains lipids, carbohydrates, okay, proteins, and several ions. Okay? So, tandaan po natin, the main component of the cytoplasm is water. Kasi kapag kayo nakakita lang sa quiz, what is the main component? Sagot ka agad, protein. Hindi pa nga natatapos basahin, protein agad. Di ba? The main component of the cell membrane is protein. Okay? Pero yung mismong cytoplasm, kasi fluid siya eh. Kumbaga, gel-like. Kumbaga, ano siya eh, para masuspend yung mga organelles po natin. It really requires water. Diba? So, basa muna tayo bago magsagot, no? Next. Ribosomes. So, konti na lang to. Okay. So ribosomes, it is composed of both proteins and rRNA, okay, that are organized into both a large and small subunit. Okay, so guys, para saan si ribosome? For protein synthesis. Okay, for protein synthesis. Okay, for protein synthesis. Okay? So, ma'am, sino po yung polyribosomes na yan? Okay? And polysome. Guys, do not forget, pag sinabi natin polyribosomes, okay, this one refers to bound. Okay? Bound ribosomes. Okay? Ma'am, anong bound ribosomes? Di ko ma'am mag-get. Guys, pag sinabi natin bound ribosomes, ito po yung nakabind sa mRNA, sa messenger RNA. Okay? So, ma'am, asan dito sa picture na to si polyribosomes? Ito si polyribosomes. Okay? Di ba magkakasama sila? Parang nakasandwich yung protein natin dito. Okay? Ito yun. No? So, yan po yun. Okay? Parang nakasandwich. Ay, hindi pala yung protein. Yung mRNA, nakasandwich siya. Okay? So, yun po. And then, we have the large subunit and the small subunit. Ito yung large. Ito si small subunit. Okay? So, guys, again, ang ribosomes, para saan daw sila? For protein synthesis. Okay, for protein synthesis. Okay, for protein synthesis. Ma'am, I thought it is the RER. Uy, ba't ba tinawag na RER ang RER? Ma'am Kasera, o bakit? Naramdaman mo? Hindi. Bakit? Because of the ribosomes. Okay, that is because of the ribosomes. Kaya nga po ang RER is also important in protein synthesis. Pareha sila. Pero sino po talaga ang gumagawa ng protein? Yung mismong ribosome. Okay? Kasi sila yung naka-attach sa mRNA. Okay? So guys, tatandaan po natin yan ha. So familiar ka na, ay polyribosomes, yun pala yun. Okay? Yung naka-sandwich doon yung mRNA. Diba? Kasi guys, tandaan po natin ha, ang importance kasi ng mRNA is for the synthesis of new protein. Diba? Or synthesis of proteins. Synthesis na, new pa yan. Synthesis of protein. Okay? So, yun po yung ibig sabihin nun mga kapatid. Okay? So, ribosomes, do not forget. Okay? Meron po siyang large and small subunit. Okay? Next. So it says ribosomes are intensely basophilic because of the numerous phosphate groups. So ano daw po ang characteristic ni ribosome? It is basophilic. Okay, it is basophilic, intensely stained with hematoxylin, methylene blue, and toluidine blue. Okay, so kung mapapansin po natin, na-stain daw po si ribosome using hematoxylin, MB, and toluidine blue. So anong color po kaya ni ribosomes kapag na-stain? Di ba? Ano kayang color? May idea po ba kayo anong kulay kaya ni ribosome kapag po yan na-stain? Meron po ba? Any idea? Intensely basophilic. Okay? Intensely basophilic. May idea po ba ng color si ribosome? Ayan. Ayan. So, sabi ni Chen, purple. Okay? Or may pagka-purple. Okay, well, actually, ang ribosomes, sometimes, no, it appears blue. Okay? Pero depende yan kasi kung anong stain mismo ginagamit natin. Kaya siya nagbumukhang purple, indigo. Wow. Okay? So, pero okay lang, no? Bluish talaga dapat siya. Pero kasi guys, sa totoo lang, saan ka naman nakakita? Sige, gato na lang. Remember H and P. Okay, H and P. Hindi ako magaling sa color ha, pero para lang mag-gets natin ito. Ang hematoxylin po kasi ay may pagka-bluish. Parang may pagka-violet o indigo, yun nga sabi. Okay? Eosine, ito yung orange or sabihin natin may pagka-red. Ayan. Okay? So, ma'am, ba't mo naman sinasabi sa amin yan? Guys, kaya minsan nag-a-appear siya o yung image natin, it appears, let's say, purple, okay? It appears pink, because of the contrast given by the H and E. May nagko-kontra kasi. O, in methylene blue, sino ba usual partner ni methylene blue? Well, usually eosine din. Diba? O, eosine and crystal violet. O, example lang yun, ha? Hindi ka pwedeng hematoxylin tapos methylene blue. Walang contrast yan. Okay? So dahil dito, ayan, nagkaroon tayo ng idea, ganun pala yun. Okay? Kaya pala may pagka-violet yung mga nakikita namin sa slides, di ba? Or sa mga pictures. Okay? Yung mga previous na binibigay ko sa inyo na pictures, makikita ninyo, bakit ganun yung color nila? Bakit may pagka-pink? Bakit may pagka-ganito? Ayan. So, yun po yung ibig sabihin. So, intensely basophilic. Kasi baka mamaya maghanap ka, ma'am, di naman siya mukhang basophil. Okay, what is the meaning of basophil? Basophilic. Diba? Guys, huwag ka maghanap ng mukhang basophil. Ma'am, question. Okay? Acidic po ba si ribosome or alkaline? Ano sa tingin niyo? Is it acidic? Okay? Or alkaline? Do you have any idea? Meron po ba, mga kapatid? Wala. Okay, may alkaline. Okay, so sabi alkaline daw. Sige, alkaline. Okay. 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 Ba't natin nasabing alkaline? Ayan, nagsabi, acidic. Diba? Nag-o-oppose sa inyo. Okay, pero marami nagsabi, alkaline. Okay? Oh, yung iba, how about the others? Meron po ba? Acidic. Ayan, nagsisimula na ang lumabas ng mga acidic na yan. Okay? Okay. So guys, kung nalilito, kulay blue, ma'am. Kaya sinabi niya na alkaline or alkaline because of the color blue. Okay? O guys eto yan Klaro natin ang mga bagay-bagay, mga kapatid, mga kaibigan. Huwag na tayong lumayo. Mag-basophil na nga lang tayo. Okay, kay basophil. Ayan. Lagi ko to sinasabi kasi gusto ko na iintindihan po natin. Ito po si basophil. Okay? Love daw niya si basic. Diba? Yan ang ibig sabihin ng basophil. Okay? So, anong connect dyan, ma'am? Remember, opposite attracts. Like ko sinasabi to, opposite attracts. So, sino ang love niya? Si basic. So, ano siya? Acidic. Okay? Kasi hindi siya maa-attract sa kaparehan niya. Diba? Ganun lang kasimple. O, edi ma'am, ang eosinophil. O, ano ang eosinophil? So, eosinophil. Edi ibig sabihin, ma'am, yung eosinophil, basic. Kaya na ba eo? Acid. Diba? Acidic yan eh. Ay, acidic. Acid ang eosin. Kaya siya naa-attract sa basic. Ganun. So ito guys, itong ribosome na to, balik na tayo sa ribosome Itong ribosomes na to, sila ay acidic in nature. Kaya sila naa-attract kay hematoxylin, methylene blue, and toluidine blue. Kasi itong mga ito ay basic. Kaya nga tinawag na basophilic. Okay? Yun po yan mga kapatid. Para maliwanag po sa atin yan. So polyribosomes attach to endoplasmic reticulum and undergo exocytosis from secretory vessel. Okay? So ayan, ito na yung sinasabi natin na mag-a-attach si ribosome kay endoplasmic reticulum para ka makabuo ng RPR. Okay? So rough, hindi po ribosome, endoplasmic reticulum yun rough endoplasmic reticulum. So next. Okay, so familiar na tayo kay ribosomes. Okay. Alam mo na okay, for protein synthesis, may large unit, large subunit and small subunit pala sila. Tapos, iniipit nila si mRNA during the formation of protein. Ganun pala yun. Diba? Okay? Now, anong meron kay endoplasmic reticulum? Okay? This network extends from the surface of the nucleus to the membrane, okay, and encloses a series of intercommunicating channels and sacs called cisternae. Okay? So, ma'am, ulit po, ang endoplasmic reticulum daw, okay, extends from the surface of the nucleus. Ibig sabihin, lagi siyang malapit sa nucleus. O, tandaan ninyo yun, Okay? Extends from the surface of the nucleus. Okay? To the membrane and encloses a series of intercommunicating channels and sacs called cisternae. So, itong mga folds na to, yung mga mukhang folds na yan, okay? Sila po yung mga intercommunicating channels. Okay? Sila po yung mga sacs. Okay? Sila yung tinatawag na cisternae. Guys, do not be confused with cisternae and cristae. Okay? Ito ay cisternae. Endoplasmic reticulum, cisternae. Do not forget, malapit sa nucleus ang endoplasmic reticulum. Do not forget. Do not forget. Next. Hindi pala malapit, katabi. Sorry. So, endoplasmic reticulum. Para saan si endoplasmic reticulum? Okay, para saan? Now, I want you to remember, guys, kapag sinabing endoplasmic reticulum, okay, we have here the different functions. No? Para saan sila? For synthesis. Okay? Remember, we have two types of endoplasmic reticulum, the SER and the RER, the smooth endoplasmic reticulum and the rough endoplasmic reticulum. Okay? Tandaan po natin, ang SER is the site for what? Lipid synthesis and carbohydrate metabolism. While the RER, the rough endoplasmic reticulum, synthesizes proteins for secretion. So protein ang sinisynthesize. Incorporation into the plasma membrane and as enzymes within the lysosomes. So in short, ang SER for lipid synthesis and carbohydrate metabolism. Ang RAF, because of the presence of ribosomes, ayan, okay, para saan daw sila? Synthesis of proteins. Okay? Transportation. So guys, ano kaya ita-transport ni endoplasmic reticulum? Okay? It moves molecules through cisternal space from one part of the cell to another. Okay? Kaya nga magkakadikit yung mga cisternae na yun. Eh. Kaya nga intercommunicating. Di ba? Sequestered away from the cytoplasm. Okay? And then storage. It stores newly synthesized molecules for storage. Okay, imagine mo ang endoplasmic reticulum, simula yata nung high school ka, okay, hanggang nung first year ka, ang nasa isip mo ay ang SER at saka RER for synthesis lang siya. Guys, hindi. It is also important in the storage of the synthesized molecules. Okay? For detoxification, okay? For detox, SER detoxifies both drugs and alcohol. Okay? So, ibig sabihin, tandaan po natin to, without your SER, hindi ka makakapag-detoxify ng mga drugs na iniinom mo. I'm not saying yung mga, yung mga abuse drugs. Yung drugs talaga na medication and alcohol. Okay? So, take note. So, para saan si endoplasmic reticulum? For synthesis, transportation, storage, and detox. Okay? Now, endoplasmic reticulum. Pa rin tayo mga kapatid. Remember, for synthesis, we have the RER and the SER. In RER, okay, dito sa RER na to mga kapatid, it is consist of sac-like as well as parallel stacks of flattened cisternae. Okay? It has highly regulated system to prevent non-functional proteins being forwarded to the pathway for secretion or to other organelles. So guys, consist of sac-like as well as paralleled stacks of flattened, okay, flattened cisternae. Ayan. Ito siya. RER, flattened. Okay, ito siya. Itong mga may bilog-bilog na yan. Okay, ito po yung smooth. Ang smooth kasi parang tubular. Okay? Yun ang difference nila, ha? Para kahit di nyo kabisado, at least sabihin niyo, kunwari, picture ng cell. Diba? Ma-identify mo, ay, alam ko to malapit sa nucleus. Okay, kilala ko to, nabanggit to ni ma'am. Ayun. Diba? Kilala mo siya. Now, Okay. Endoplasmic reticulum pa rin po tayo. Now, kung mapapansin po natin, we have here your polyribosomes. Okay? And the ER-bound polyribosomes, we have the free polyribosomes. So, kilalanin niyo to, di ba? Ito yung ribosome natin. Okay? Yung may large subunit and small subunit, natatandaan ninyo, di ba? Tapos ito yung mRNA natin. Okay? And then, dito naman, okay, we have the ER-bound polyribosomes. So, ibig sabihin, ito daw yung polyribosome na naka-attach kay endoplasmic reticulum. Okay? Doon sa may sister May, remember? Okay. Now, magsimula po muna tayo kay free polyribosome. So, what will happen to a protein, okay, that will be synthesized, okay, by the polyribosomes? Kasi, guys, di ba, yun naman talaga ang purpose ng ribosome natin eh. It is engaged in the formation of protein. So, pag-usapan natin, paano ba na-form? Paano ba nabubuo yung mga proteins na yan? Okay? So, ma'am, ano po yung dapat kong tandaan dyan? So, let's start with this one. The proteins that are synthesized by the ribosomes, tandaan po natin, yan ay pwedeng magamit, okay, sa cytosol or sa cytoskeleton. Okay? Yun daw ang purpose niyan. Number 1. Yun yun. And then, some proteins will be imported to mitochondria, peroxisome, and the nucleus. Okay? So, yung iba sa organelles mapupunta, yung iba sa mismong cytosol. Okay? Or cytoskeleton. Now, how about the misfolded and denatured proteins? Guys, gusto ko lang pong i-highlight ito eh. Gusto kong maintindihan natin na hindi naman, parang QC. Alam niyo yung QC, yung quality control. Okay? Hindi po lahat perfect na nagagawa ng katawan mo. Okay? Hindi lahat perfect. Okay? There are some proteins that are misfolded and denatured. So, in short, pangit. Diba? Kumbaga sa quality control, sabi, hindi to pasado sa quality control. Okay? That's why ubiquitin is important because those proteins which are misfolded and denatured will conjugate, okay, or will bind to ubiquitin. Okay? So, ma'am, sino ba si ubiquitin na yan or ubiquitin? Guys, alam niyo po ba si ubiquitin, okay, it is for the degradation of proteins. Degradation. Okay, for degradation. Okay, for degradation of proteins. Okay, for the degradation of proteins. Ubiquitin. Okay, ma'am ulit po, for degradation of proteins. Sino daw si ubiquitin? Kasi guys, anong gagawin mo sa mga misfolded or denatured proteins? Wala. But they are made up of proteins. That's why kailangan nila si ubiquitin. Okay? And then it will undergo degradation process. Okay? Tandaan po natin yan, ha? Mga kapatid. Next. So, familiar ka na, ganun pala nangyayari sa mga free polyribosomes. Yung mga proteins nila mapupunta sa mga organelles or sa mismong cytosol para magamit. Yung mga misfolded or mga denatured, okay, magbabind sa ubiquitin para masira sila kasi wala naman na silang kwenta. Ganon. Diba? Ma'am, how about for the ER-bound polyribosomes? Okay, ER-bound polyribosome. So, ito yung sa RER ang pinag-uusapan natin. Remember, ito po ngayon yung mapupunta sa Golgi apparatus. Okay? And remember guys, ang Golgi apparatus or the Golgi body is the packaging site of the cell. Okay? Kaya nga, it will process and sort. Okay? And then, saan mapupunta? Once na na-process na, okay? Or na-sort na. O, eto for lysosomes. Okay? O, eto, punta sa secretory vessels. And then, kapag nandun na sa may secretory vessel, okay, dyan natin malalaman if the proteins will be secreted from the cell, okay, or proteins, or mapupunta siya sa proteins of the cell membrane. Kasi remember, ang cell membrane, ang main component niya is protein. Okay? Or kailangan ba palabasin natin to? Kailangan ba mag-undergo to ng exocytosis? Meron bang ibang nangangailangan? Padala mo doon ngayon. Ganun ang ginagawa ni Secretary Vesicle. Okay? So, guys, may idea ka na. And then, meron ulit mga denatured or misfolded proteins. Still, magbabind sa ubiquitin for degradation process. So, ganun po nangyari sa endoplasmic reticulum, mga kapatid. Okay? Ganun po ang nangyayari sa, tawag dito, sa proteins. Okay? Na binubuo or nabubuo sa mga cells natin. Okay? So, next. Ayan, did you know osteogenesis imperfecta? Okay? In osteogenesis imperfecta, the bone cells synthesize and secrete defective collagen molecules which cannot assemble properly and produce very weak bone tissue. Eh guys, ang collagen is protein. Di ba? Ang collagen ay isang protein. Eh guys, kung protein yan, mayroong gumawa sa kanya. Remember. Okay? So, ibig sabihin, okay, yung endoplasmic reticulum, during the formation of this collagen, okay, eh, ibig sabihin, hindi nasira, ni ubiquitin. Kaya naging ganyan yung tao. Kaya siya nagkaroon ng osteogenesis imperfecta. I know, dahil very active naman kayo sa social media, nakakakita kayo ng mga taong ganyan. Okay? So, tandaan po natin, okay, familiar na tayo, ay, ganun pala si osteogenesis imperfecta. Okay, imagine mo, no, osteo, so ang nasa isip mo, sino? Bone. Pero ang totoo, ang main problem nila is yung mismong collagen. Okay? Tandaan po natin yan, mga kapatid. Okay? Next. Okay, SER. Okay, SER, the cisternae are often more tubular. O, ito yung sabi ko kanina, tubular sila. And more likely to appear as interconnected channels of various shapes and sizes than as stacks of flattened cisternae. So, kino-compare niya kay RER. Diba? Kasi si RER is flattened. Okay? Tandaan natin yan. So, para saan po si SER? Okay? That is for temporary calcium sequestration and release, particularly in the sarcoplasmic reticulum. Guys, yung sinasabing sarcoplasmic reticulum, this is actually seen in muscles. Okay? Sarcoplasm, di ba? Muscle yun eh, sarcoplasmic reticulum. Pag sinabi natin sarcoplasmic reticulum, yung muscle tissue, okay, yung muscle cell, meron siyang endoplasmic reticulum. At ang tawag sa endoplasmic reticulum ng isang muscle ay sarcoplasmic reticulum. Okay? So guys, take note. Okay. Guys, familiar ba kayo dito sa picture na to? Familiar po ba kayo dyan, mga kapatid? O hindi? Ngayon nyo lang ba nakita yan? Ayan, so sabi, jaundice. Jaundice baby mom. Ayan. Okay, so very good, no? Alam na ninyo, di ba kahit hindi nyo pa talaga naiintindihan kung bakit nagkakaroon ng jaundice ang isang tao, still nakakapag-identify kayo ng mga taong jaundice. Okay? So, ma'am, ano ba ibig sabihin yan? Okay? Pag-usapan muna natin itong SER na to. Okay? SER is abundant in many liver cells. O yan, napakinggan ninyo yan. Tsaka basahin ninyo. Okay? In many liver cells. So, sa hepatocytes daw. Where it contains enzymes responsible for glycogen metabolism, for processing endogenous molecules such as component of bile, and for the oxidation, conjugation, and methylation reactions that neutralize potentially toxic exogenous molecules such as alcohol, barbiturates, and other drugs. Nung wala ko naintindihan doon, ang naintindihan ko lang, ma'am, SER. Okay? Alam mo ba kung bakit nagkakaroon ng jaundice ang isang tao? Guys, kung bakit nagkakaroon ng jaundice, that is because of the increase in bilirubin. Kilala mo si bilirubin, di ba? First year ka pa lang, kilala mo yan. These are the end products of hemoglobin degradation. Okay? End product of hemoglobin degradation. Bilirubin yun, ha? Okay, bakit ka nagkaroon ng jaundice? Kasi mataas ang bilirubin. Yan yung bilirubin, end product of hemoglobin degradation. So what? Ano connect? Ano connect ng SER? Okay? Guys, kasi ang SER is very important. Bakit? Kasi anong sabi dito? Okay? For conjugation. Okay? For conjugation. O, ito yan. It contains enzymes. Yun na lang i-highlight natin eh. Huwag na nating buuin lahat eh. Okay? Yun na lang. Meron siyang enzyme and it is an important site for the conjugation. Okay? O, yun. Ma'am, ano ibig sabihin? Very simple. Natatandaan po ba ninyo sana natatandaan ninyo. Nagplebo naman kayo eh. So, naiintindihan ninyo to mga kapatid. Kahit pa paano. Meron po tayong tinatawag na B1, B2. Sa bilirubin. Yung direct, mali. Indirect pala to. Indirect. Ito si direct. Okay. So, insoluble. Lagay ko na lang dito. Soluble. Unconjugated. Okay, ano na lang. Ito yung conjugated. Okay, so B1, indirect, insoluble, unconjugated, pare-parehas yan. B2, okay, direct, soluble, conjugated bilirubin. Yun ang ibig sabihin yan. Direct bilirubin is B2, indirect bilirubin is B1, insoluble bilirubin is B1. Okay, conjugated bilirubin is B2. Yun yan. Synonym nila yan. O ma'am, anong connect? O yun. Hey guys, diba may nakalagay dito conjugation. Conjugate, unconjugated. Okay? Saan daw ginaganap? Saan daw nangyayari yung conjugation process na yan? Sa SER. Okay? Hey guys, ganito kasi yan. Si B1, sabi natin, it is insoluble. So in short, hindi yan maiihi. Hindi mo rin yan maitatain. Naintindihan niyo po ba ibig kong sabihin? Dahil insoluble sila. Okay? Hindi sila mabilis mahalo sa iba. Di ba? Hindi siya soluble eh. So ma'am, ano ibig sabihin? Si B1 dapat ma-convert sa B2. Bakit? Because B2 is soluble. And B2 is conjugated. Ayan. O ma'am, anong ibig sabihin? Okay? So, kailangan daw ma-conjugate ni B1 para maging B2. And remember, anong enzyme? Okay? Anong enzyme ang kailangan natin for the conjugation of B1? Ito yung tinatawag nating UDPGP. Okay? Uridyl diphosphate glucuronil transferase. Di ba? So, iba dito, familiar na. Uridyl diphosphate glucuronil transferase. Uridyl diphosphate glucuronil transferase. Okay. So, ma'am, ano yan? O, di nakita mo na may enzyme responsible. Okay? May conjugation. Okay? So, ano ibig sabihin? Bakit daw nagkaroon ng jaundice? Kasi hindi nagkaroon ng conjugation ng B1, okay, to B2, or maaaring naipon yung bilirubin mo. Yun ang ibig sabihin yan, mga kapatid. Kung bakit tayo nagkakaroon ng jaundice. Okay? Kaya nga kapag nasira ang liver cells or ang hepatocytes, kakainom ninyo ng alak, ng alcohol, guys, remember, alam mo kaagad, jaundice. Okay? So, I hope guys, it makes sense. No? Next. Golgi apparatus. Konti na lang. So, Golgi apparatus, functions, packaging process, lysosome formation, and then glycosylation, phosphorylation, and limited proteolysis of proteins. Okay? So, packaging process. Natatandaan ninyo yung sa ER bound na polyribosomes, yung nasa RER, yung na-form down na protein doon, guys. Remember, ipapackage daw yun. Okay? Natatandaan ninyo. Tapos may sorting pa nga. Natatandaan. So, guys, kung hindi, balikan po ninyo yun. Okay? May copy kayo, ha? And then, glycosylation, phosphorylation, and limited proteolysis of proteins. Kaya nga diba ang Golgi apparatus is what? Golgi apparatus is the packaging unit or the packaging site of the cell. Okay. So, guys, kung mapapansin po natin, we have here the RER. Okay? We have the flattened cisternae, diba? Ito yung mga ribosomes po natin. Then it forms a vesicle to transport what? To transport your proteins. Okay? And then ipapackage daw ni Golgi apparatus para alam niya kung saan niya dadalhin. Diba? So, yun po yun. So, take note. Next. Lysosomes. So, lysosomes are the site of intracellular digestion and turnover of cellular components. Guys, kaya po, di ba tinatawag na lysosomes, these are the suicide band or suicide bands of the cell. Okay? Kasi sila yung responsible for the digestion. Okay? So, abundant in cell with great phagocytic property. So, yung mga phagocytes daw, mas madami raw silang lysosomes compared sa mga hindi phagocytic. Then, it contains hydrolytic enzymes with a pH 5.0. So, ibig sabihin, acidic yung environment ng lysosome, okay, for the digestion. Okay? It has acid phosphatase. Okay? ACP is acid phosphatase, di ba? Okay? It contains acid phosphatase, ribonuclease, protease, nuclease, phospholipase, catepsin, sulfatases, lipases, and the beta-glucuronidase. Okay? So, ma'am, ma'am, hindi dapat masira yung lysosomes sa loob, ma'am, ng cell. Bakit? Ma'am, kasi ma'am, kasi masisira yung cell. Bakit? Kasi ma'am, may hydrolytic enzymes, ma'am. So masisira po yung ibang laman ng cells, ma'am. Kaya hindi po pwedeng mag-leak yung mga enzymes na yan. You know what, guys? Okay? Tandaan po natin to. That the pH of your cytosol, ayan ha, tandaan niyo. The pH of your cytosol is 7.2, which means, okay, these hydrolytic enzymes will become inactive. Okay, magiging inactive sila once na napunta sila doon sa environment na hindi appropriate para sa kanila. So I want you to remember that. Okay? Kaya huwag kayong matakot na, hala, baka masira yung hydrolytic enzymes, tama, baka masira yung mga hydrolytic enzymes ko. Guys, hindi. Magiging inactive lang naman sila. Okay? Magiging inactive. Okay? So, tandaan niyo yan. So, next. So, we have here the autophagy. Okay, autophagy, guys. Control digestion of aging intercellular structures. Okay, to form autophagosome. So, ano ibig sabihin? Lahat ng components ng cells po natin tumatanda. Okay, tumatanda yan. Kaya ano ang sinabi dito? May autophagy. Control digestion of aging intercellular structures. Okay? Digested products are recycled. Imagine mo, nire-recycle yung digested products, okay, by the cell to permit renewal. Ayun naman pala. Akala ko ni-recycle lang. Yung pala, for renewal. Rearrangement and reconstruction of the cytoplasmic content. Okay? So kung titignan natin, ay, ganun pala ang autophagy. Maganda pala siya kasi yung mga pangit na parts ng cell, okay, or yung mga structure sa loob ng cell, i-recycle niya for renewal, reconstruction, and rearrangement. Okay? So, tandaan po natin yan, ha? Next. Hetero. Okay? So, heterophagy, digestion of materials that will enter the cell like bacteria, virus, and fungi. Okay? Ma'am, ano ibig sabihin? Hetero. Ibig sabihin, mga kakaiba, outside. Di ba? So, from the outside environment. Okay? That is for heterophagy. Okay? One of the stages of phagocytosis were in lysosomes fused with the bacteria forming a phagosome. Okay? So, guys, remember sa phagocytosis, di ba? Merong vesicle formation kasi pumasok siya sa isang lysosome, nag-engulf, kaya siya tinawag na phagosome. Okay? Take note. Okay, as in take note. Heterophagy, for foreign antigens po to. Okay? Bacteria, virus, fungi. Okay? Ayan. So, I think na-discuss ko na naman to kanina. So, skip ko. Ayan. So, we have here the different abnormalities associated with the lysosomes. Okay? So, kasi alam na natin na ang lysosome ay marami siyang mga hydrolytic enzymes. And, alam nyo ba na konti lang yung binigay ko sa inyo, marami pang klase. Okay? So, we have here the Hurler syndrome 1. Okay? Which has a faulty enzyme known as the alpha. This is alpha. Hindi kasi malagyan ng symbol eh. Alpha L hydronidase. Okay? Alpha L levo, diba? Hydronidase. Now, the main organ affected here in Hurler syndrome is the skeleton and the nervous system. In McArdle syndrome, okay, the faulty enzyme is the muscle phosphorylase. So, skeletal muscle po yung affected. In Tay-Sachs syndrome, okay, in Tay-Sachs syndrome, the faulty enzyme is the GM2, okay, gangliosidase, which affects the nervous system. Gaucher, okay, favorite ko to, highlight natin yan, tsaka ito. Okay, in Gaucher, or Gaucher's disease, the faulty enzyme is the glucocerebrosidase, and the affected organ is the liver, or the liver and spleen, and the eye cell disease. Okay, eye cell disease, the faulty enzyme is phosphotransferase for M6P formation. Okay, so the affected organs are the skeleton and the nervous system. Pero guys, ulitin ko, ang paborito ko dito ay si McArdle and si Gaucher. Si Tay-Sachs din pala. Yan. Yan mga paborito ko dyan. Madalas kong tanong kung anong enzyme or anong disease. Yan ang lagi kong tanong. Okay? So, next. Mitochondria. Okay, now, in the mitochondria, membrane-enclosed organelles with arrays of enzymes specialized for aerobic respiration and production of ATP with high-energy phosphate bonds, which supplies energy for most cellular activities. Anong ibig sabihin? Mitochondria is the powerhouse of the cell. Okay, that's the powerhouse of the cell. Okay, so anong ginagawa daw ni mitochondria? It sequesters calcium ions in the form of calcium phosphate granules. Okay, aside doon sa taga-supply siya ng energy. Okay, so do not forget. Next. Okay, now what are the parts of the mitochondria? We have the porine, okay, or the outer membrane contains porine. Inner membrane contains cristae. Ito yun ha. Hindi cisternae. Iba yun. Intramembranous space and the matrix. Okay? Matrix, which contains calcium and magnesium. Okay? Ayan. So, familiar ka, ganun pala yun. Yung pala yung sinasabing mga parts ng mitochondria. Meron siyang cristae. Okay? inner membrane contains cristae. Ito yung mga fold sa loob. Okay? Yan yun. Okay? Outer membrane. Okay? And then the matrix, yung kung nasaan po yung mismong fluid. Okay? And ito daw, matrix na to ay may lamang calcium and magnesium. Okay? And then the outer membrane contains porine. Okay? May mga porins yan. Pero dito walang nakalagay. Pero may mga porins. Ibig sabihin may mga pores. Okay? So next. So peroxisomes. Ayan. So guys, no, parang mahaba-haba na ang ating nag-discuss. Pero tatapusin natin ito hanggang mamayang hapon. Peroxisomes. Spherical organelles enclosed by a single membrane and need for their enzymes producing and degrading hydrogen peroxide. Okay? O guys tandaan po natin yan. Spherical organelles enclosed by a single membrane and need for their enzymes producing and degrading hydrogen peroxide. Okay? The enzymes inactivate various potentially toxic molecules, including some prescription drugs, particularly in the large and abundant peroxisomes of the liver and kidney cells. So guys, imagine, di ba? Kanina, lysosome lang ang pinag-uusapan natin, and yet now we have these peroxisomes. Okay? So, parehas po silang may enzymes. Okay? Actually, yung endoplasmic reticulum mo nga, meron din naman silang enzymes. Remember, may example na tayo kanina. Okay? So, ma'am, edi ibig sabihin, marami pong peroxisomes sa liver and kidneys. Yes. Bakit? Kasi guys, yung metabolism ng drugs, normally sa liver. Okay? And then for the excretion, kailangan mo ng kidney. Kaya kailangan po talaga ng peroxisome sa mga areas or sa mga organs na yun. Okay? Ayan. So, ma'am, ano yung mga yan? Okay? Guys, tatandaan niyo po si endoplasmic reticulum. Okay? Si endoplasmic reticulum, ito po, nakikita niyo po, we have the pre-peroxisomal template. Ano ibig sabihin? Nag-start ang formation ng peroxisome from the endoplasmic reticulum. Okay? Ma'am, ulitin natin. Okay? Ang peroxisome, saan galing? Sa mismong endoplasmic reticulum. Okay? Mang-ulit po, ang peroxisome, saan galing? Sa mismong endoplasmic reticulum. Gusto kong i-highlight yun sa inyo. Okay? So, take note. And then, the names of your peroxisomes will depend on the enzyme, okay, inside that peroxisome. So, maraming pangalan po ang mga peroxisomes. Okay? Okay, huwag tayo munang mag-break. Pasensya na. O sige, mag-break na nga kayo. Stop ko Guys, ang time sa akin, 9:35. Okay? Resume tayo ng 10 para medyo mapahinga naman kayo. Okay, 10. Yung iba, pwede pong mag-leave. Okay, so cytoskeleton. Now, I want you to remember, okay, pag sinabi po natin cytoskeleton, it provides structural support to the cell. Okay, so madaling tandaan. It stabilizes junctions between the cells. It plays an important role in the movements of organelles and cytoplasmic vesicles. Allows movement of the entire cell entire cell. Tanggalin natin to. And during the cell division. So guys, mamaya po mag-discuss po tayo ng cell division. Okay, so kaya po importante po itong topic na to, no? I mean, itong mga parts na to. A complex array of microtubules, microfilaments, and the intermediate filaments. So guys, do not forget, pag sinabi pong cytoskeleton, okay, number one, it provides structural support to the cell. Okay, and it makes the cell more stable. Okay, what else? Guys, tandaan po natin that your cytoskeleton is very important in a way that it allows movement, okay? It allows movement without any destruction of any parts or any component of the cell. Kasi yung iba, di ba, masyadong rigid. Okay? So kapag masyadong rigid po ang cell, guys, konting galaw lang, may masisira, di ba? But because of the cytoskeleton, again, it allows movement. Okay? So pag-usapan natin si microtubules, microfilaments, and the intermediate filaments. So again, we have the microtubules, microfilaments, and the intermediate filaments. Okay, so ma'am, paano po yan? Masyadong complex yata mamang itsura. So tignan natin. Guys, we have here the table, okay, for the complex array of cytoskeleton, the microtubules, microfilaments, and the intermediate filaments. I am 100% sure that you can memorize this table. Pero gusto ko lang pong i-highlight. Okay? Gusto ko lang pong i-highlight yung iba. Okay? So ano po yun, ma'am? Do not forget, for microtubules, okay, the subunits are the heterodimers of the alpha-beta tubulin. Okay? In microfilaments, the subunit is the G-actin monomer. When we say G, it means globular. Okay, globular. And in the intermediate filaments, we have here the subunit antiparallel tetramers of two rod-like dimers. Okay, so ano dito yung tatandaan ko? Itong dalawang paborito ko. Okay, the subunit of microtubules and the subunit of microfilaments. So, ina-highlight ko lang po, no, for our activity mamaya. Okay. The overall structure, microtubules, hollow tube. Kaya nga tinawag na microtubules. Okay. With a wall of 13 parallel protofilaments. In microfilament, two intertwined filaments of F-actin. Okay, so two intertwined filaments. While for the intermediate filaments, cable of four intertwined protofibrils, each consisting of bundled tetramers associated end to end. Okay. For polarity, of course, same po si microtubules and microfilaments that it has positive and negative polarity at both ends. Unlike sa intermediate filament, no apparent polarity. Okay. For the relative stability, dynamic, in cytoplasm, stable in axonins sa microtubules. Okay? And for intermediate filament, it is stable. Key functions, ito pag-usapan lang natin to. Microtubules, it maintains cell shape and polarity. It provides tracks for organelle and chromosome movement. So, move cilia and flagella. So, ito yung mga apical structures po natin, di ba? Yung cilia and flagella. So, microtubules, it allows movement. Okay? It maintains the shape and the polarity of the cell. Okay? And it moves cilia and flagella. Microfilaments, it contracts and moves cells. Okay? It contracts and moves cells. Change cell shape. Okay, so nagbabago ha. Kanina, minemaintain po ni microtubule yung shape. Pero dito sa microfilament, it changes the shape of the cell. Okay? It is also important in cytokinesis. Ano ba ibig sabihin ng cytokinesis? Cytokinesis means cytokinesis. This is the cytoplasmic division. Okay? Cytoplasmic division of a cell. Yan po yung nangyayari at the end of mitosis or meiosis, cytoplasmic division. So, importante daw ang microfilament in cell division because of the cytokinesis. So, cytoplasmic transport and streaming. With microfilament, ha? And then, intermediate filament, of course, the functions are to strengthen cell and tissue structure. So, more on strength. Kaya pala stable talaga siya. Okay? It maintains the cell shape, okay, which is similar with your microtubules. Plus, it maintains the nuclear shape. Hindi lang siya doon sa mismong cell membrane. Even the nuclear shape, okay, tandaan po natin, minamaintain po ni intermediate filament yung shape. Okay? Centromeres. Anong meron kay centromeres? Okay, a zone of cytoplasm usually centrally located in the cell adjacent to the nucleus. So, again, adjacent, okay, to the nucleus, katabi. Okay? A microtubule organizing center for the mitotic spindle. Kasi malapit na natin i-discuss si cell division. And consists of paired centrioles. Contains a pair of centrioles. Ayan. Center of activities associated with cell division. So, i-highlight natin tong part na to. Center of activities associated, okay, with cell division. Okay. So, ma'am, anong ibig sabihin yan? Okay? Tandaan po natin, mga kapatid, meron daw tayong centrioles. Okay? We have two centrioles. Okay? We have two centrioles. So, ma'am, ano ibig sabihin yan? Itong centrosome na to, which is pinag-uusapan natin, okay, ay mayroon daw siyang centrioles. Okay? So, pair of centrioles. Ano ibig sabihin? Dalawang centrioles meron po tayo sa isang centrosome. Okay? At iyan pong centrosome na yan ang nasa tabi ng nucleus. Okay? So, tandaan po natin, this is the center of activities associated with the cell division. And kung mapapansin po natin, hindi parallel, okay, yung ating centrioles. Ang itsura niya para siyang naka 90 degrees. Okay? So, do not forget. So, ito po yung ating centriole. Oh, guys, huwag na kayo magtaka. Ma'am, centrosome tapos centriole. Okay? Kasi nga, ang centrosome ay mayroong dalawang centriole. Diba? Yung naka 90 degrees, remember? So we have triplet microtubules. So ito po yung mga microtubules natin. Okay? And itong triplet microtubules na yan, ilang beses daw yan umulit? 1, 2, 3, 4, 5, 6, 7, 8, 9. So meron po tayong 9 triplet microtubules per centriole. 9 triplet. Naintindihan niyo naman siguro ibig ko sabihin. 9 na beses to. Okay? Take note. Next. The centrioles are also known as diplosome. Highlight. Okay, kilala mo na si centriole, yung nasa loob ng centrosome. Si centrosome na nasa tabi ng nucleus. Okay? So centrioles, self-duplicating organelles po sila. So i-highlight natin din yan. Okay? So again, centrioles are the self-duplicating organelles, and it is also known as the diplosome. Okay? Now, it says here that centrioles are organelles which are only active during the cell division. So mamaya pag-uusapan natin yan. Ito po yung mga centrioles natin. Okay, so mamaya pag-uusapan natin yan. So, ito po yung sa microfilament. Okay, ito po yung itsura ni microfilament. So, anong sabi ng sa microfilament? Okay, sa microfilament, two intertwined filaments of F-actin. Okay, so ito po yun. Diba? Two intertwined. Okay, ganyan po itsura niya. Next. Now, for the intermediate filaments, okay, intermediate filaments, so we have different classes, no? Or classifications. 1, 2, 3, 4, 5, and 6. Okay, so ito guys, nice to know. Lagyan ko ng nice to know. Okay. So, gusto ko lang pong i-highlight lang dito, although nice to know to ha, ibig sabihin hindi ko siya masyadong binibigyan pansin sa quiz or exam. So, again, we have here the different classifications, 1 to 6. So, in class 1, okay, the protein associated is the acidic cytokeratin. And this protein is distributed in different epithelial cells. Okay? So, ganun din kay class 2, basic cytokeratin, okay, epithelial cells, and so on. Okay, so ito po, nice to know. Inuulit ko ha, nice to know. Okay, and remember guys, ano ba ang trabaho ni intermediate filament? Okay, it strengthens the cell. Okay, what else? Sabi natin kanina that your intermediate filaments maintains the cell shape also. Okay, and ano pa? Hindi doon nagtatapos yun. Okay, it maintains the nuclear shape. Okay, so take that. Konti na lang. Cytoplasmic inclusions. So, ma'am, anong meron sa cytoplasmic inclusions na yan? In the cytoplasmic inclusions, we have here the fat droplets. Okay? the glycogen granules, and the lipofuscin. Now, I want you to highlight, okay, the fat droplets for the accumulation of lipids. Okay, glycogen granules, the aggregates of carbohydrate monomer. So, ang glycogen, guys, normally, we use a specialized stain, which is the periodic acid sheet. Periodic acid sheet. Okay? Para maging visible po yung glycogen natin. And then we also have here the lipofuscin, which is the wear and tear pigment, which is derived from the residual bodies after lysosomal degradation. So kapag pala may lipofuscin sa isang cell, it means, okay, that there is a degradation, that there is a lysosomal digestion that occurred. Okay, so lipofuscin. So again, these are just some of the cytoplasmic inclusions, hindi ito kompleto. Okay, some lang po ito. So we have here the fat droplets, glycogen granules, and the lipofuscin. Do not forget your periodic acid sheet and the wear and tear pigment. Next, nucleus. Medyo bibilisan ko na ha. The nucleus, the most prominent structure within the cell. That is correct, di ba? And that is very obvious. Guys, ang nakikita mo lang talaga sa isang cell ay si nucleus. Okay, plus the periphery of the cell membrane. Found in all cells except for mature erythrocytes and blood platelets. That's very obvious. Usually seen singly in one cell except for skeletal muscle cells and osteoclasts. So, ano ibig sabihin? The skeletal muscle cell, okay, and your osteoclasts are what? Multinucleated. So, sila lang po ang naiiba sa lahat. Skeletal muscle and osteoclast. Parehas pong multinucleated. Don't forget. It has an affinity to basic dyes. Ayan na naman tayo. Because it contains DNA. Alam na ninyo ibig sabihin yan. Composed of nuclear envelope, nuclear pore, nucleolus, nuclear matrix, and chromatin. Okay? So alam na ibig sabihin ng affinity to basic dyes. Okay. So ito po guys, meron po tayo dito tinatawag na mga nuclear pores, di ba? The nuclear envelope, okay, the chromatin, the nucleolus, ayan, okay, the nucleoplasm, okay, the outer membrane, the inner membrane. So guys, hindi na po natin iisa-isahin iyan. And I know you are very much familiar with the cell structure. Okay? Alam ko high school pa lang kayo naaral niyo na to. So now we have here the nucleoplasm. Okay, or the chorioplasm. Ang babanggitin ko lang po yung mga importante. It is a semi-fluid, a colloidal solution to which the chromatin material and nucleolus are suspended. Madali lang. Serves as medium for the diffusion of metabolites and larger molecules. Okay? Madali lang. Next, nuclear envelope. Anong sabi dyan? It forms a selectively permeable barrier between the nuclear and the cytoplasmic compartments. So, obvious yun. Lamin is the major component which binds to membrane proteins and associate with chromatin in non-dividing cells. Okay? So, tandaan niyo yung lamin yan, a major component which binds to membrane proteins. Okay. Nucleoporins regulate the movement of macromolecules, and the energy for transport is derived from guanosine 5-triphosphate with specific GP phase, helping provide directionality to the transport. So, sino po ang tatandaan natin dyan? Si lamin, okay, and si nucleoporin. So, lamin, major component, okay, and then nucleoporin regulate the movement of macromolecules. Now, may papakita ko sa inyo. Si progeria. Now, I guess guys, lahat kayo halos mahilig kayong tawag dito, browse sa social media, and then makikita ninyo may mga different diagnosis, then may mga instances, I think sa kapuso mo yata yun, kapuso mo Jessica Soho yata yun, yung may progeria. Yes, yan, correct. Yung ano to, ayan, yung 16 ma'am na mukhang matanda, ayan, sa FB talaga. Ayun. Well, actually nakita ko siya sa, saan nga to? Sa kapuso mo. Yun. Okay? Pero kasi guys, ito lang yun based kasi doon sa sinabi ng doktor doon na it is a possible progeria, a type of progeria. Okay? Kasi dati, ang pagkakaalam ko lang sa progeria, may beaking of nose, beak. Yung parang, alam mo siguro yung beak, nagmumukhang tuka yung ilong. Katulad dito, ito guys, ito yung typical na progeria kasi talaga. Pero yung sinabi doon sa kapuso mo nung doktor, hindi ko po kinu-question na, hindi ako sa different types of progeria. Nung sinabi niyang progeria yun, siguro nga, kasi guys, alam niyo ba kung bakit? Kasi lahat kasi ng may progeria, merong abnormal aging. O ano sabi dito? Nuclear envelope is abnormal. Kaya nga tinawag na laminopathies. Alam mo na kung bakit siya tinawag na laminopathies. Bakit? Because the lamin is the major component. Di ba? Yun ang sabi dyan eh. So, major component which binds to membrane proteins. Okay? So, kaya nga, ang progeria is a laminopathy. Okay? Abnormality sa lamin. Okay? Which causes premature aging. Kaya alam niyo po ba, merong mga cases, most especially sa ibang bansa, ang daming cases ng progeria. Baka mamaya dito sa atin, sa Pilipinas, no? O yung iba sa inyo, wala dito sa Pilipinas. Baka doon kinasaan kayo, hindi natin alam kung meron din. But I think, no, halos lahat naman ng countries, meron namang mga different cases. Okay? So, sa ibang bansa, ang dami po talaga. Meron niya ako doon nakita nga na, ang tawag dito, 5 years old siya, pero ang bone density niya, ewan ko kung natatandaan ng iba kong mga nagsisi dito, yung bone density niya, pang 18 na. Tapos nung nag-18, nung nag-18 siya, ang bone density niya pang 5. Pang 45. Ang bone density niya Ibig sabihin, di ba dapat, kung ang edad ninyo ngayon is 20, 21, 22, ganyan ang edad ninyo ngayon, dapat, ang bone density mo is, nagmamatch dun sa edad mo. Okay? But in progeria, guys, hindi. Mas na-advance. Kaya na, magugulat ka nga, mas mukha pa siyang matanda dun sa nanay niya. Okay? Yung 16 years old. Ang ganda ng bata. Tapos biglang meron siyang progeria. Okay? Pero wala siyang beaking of nose. Yun ang gusto ko lang sabihin sa inyo. Dahil isa yun sa mga features ng mga may progeria, yung pag-beak ng nose. Okay? And then, hindi sila pwedeng madapa basta-basta. Bakit? Dahil nga yung bone density nila is hindi akma sa edad nila. So, maaaring 18 years old sila, pero ang bone density nila, pang 60 na. We cannot tell. Diba? So, parang ganun po siya. Eh, di ba mga matatanda hindi pwedeng ma-fracture kasi nga, mahirap ng ayusin. So, yun ang problem sa progeria. So, ngayon alam mo na, okay. Kanina yung sa osteogenesis imperfecta, ngayon, kanina jaundice, tapos ngayon progeria. So, nagkakaroon kayo ng idea kahit papaano. O di ba nga, na sabi nga ni DJ, ma'am, yung yung sa FB, ayan, di ba? So, ganun lang yan. Kumbaga, exposed naman kayo eh. So, dahil exposed kayo, medyo marelate nyo pa rin ito. Okay? So, next. Alam nyo niyan si progeria. Parang dati nga, mayroon pang case na parang magkakapatid sila, pero lahat halos may progeria. Pakapuso rin yata yun. Hanapin nyo na lang. So next, chromatin. In non-dividing nuclei, chromatin consists of the DNA. Ayan. So guys, alam na kung nasan yung DNA, nasa nucleus yan, di ba? And its attendant proteins is in a largely coiled state. So we have two types of chromatin, the heterochromatin and the euchromatin. Sino po si hetero? It appears as coarse, electron-dense material in the electron microscope and as intensely basophil clumps in the light microscope. O yan alam nyo na yun. Diba? Yung electron and the light microscope. Euchromatin, visible as finely dispersed granular material in the electron microscope and as lightly basophilic areas in the light microscope. Okay? So, ma'am, makikita ko po ba yan? Guys, syempre, hindi po siya ganun ka-visible. Diba? Kasi hindi siya ganun ka-visible. But I just want you to remember the heterochromatin and the euchromatin. Okay. The dispersed euchromatin contains, euchromatin to contains regions of the DNA undergoing active transcription and is more prominent in metabolically active cells. Okay? So, contains regions of the DNA undergoing active transcription. So, talagang very important po itong ating chromatin. Okay? Kapag may cell division. Okay? Kaya nga DNA na pinag-uusapan dito, di ba? Okay? Kaya pag nagkaroon po ng problem po sa DNA, ang unang maapektuhan is the cell division. DNA in chromatin is extensively packaged by associating with basic proteins called histones. So, sino daw si histone? Siya po yung basic protein. Okay? And with various non-histone proteins. Okay? The structural unit of DNA and histones is the nucleosome. So, sino si nucleosome? That is the structural unit of DNA and histone, which has a core of 8 small histones around which is wrapped DNA with about 150 base pairs. Parang wala kang naintindihan doon, ano? Pero tignan na lang natin ito. Okay? So, we have here the DNA. Ayan. Okay? The nucleosome. Okay? So, sino daw si nucleosome? Di ba? This is the structural unit of DNA. Structural, yun ang sabi natin kanina, structural unit of DNA and histones. So, nasaan si DNA? Ito si DNA. So, ito si DNA. Ito ang histone. Okay? So, what else aside from that? Sabi dito, packed nucleosomes, ayan, in 30 nanometer chromatin fiber. So, pinak. So, kaya sila nagdikit-dikit. Okay? And then, take note, meron po tayong extended loops of transcriptionally active chromatin. Okay? Tethered to protein scaffold. Okay? Then dito nakalagay condensed heterochromatin and dispersed eurochromatin. Euchromatin. So ito po yung hetero, parang hindi siya, parang very irregular yung coiling po niya. Okay? Pero makapal. Unlike in euchromatin, okay? Ang euchromatin medyo hiwa-hiwalay. Okay? Ang heterochromatin, kaya nga siya nagiging intensely basophilic dahil doon sa pagkakaklump niya. Okay? Or doon sa pagkakadikit-dikit niya. Okay? So, that's the difference. Para lang po maintindihan lang po natin. Ayun pala si hetero tsaka yung si euchromatin. Okay? So, do not forget the nucleosome. Okay? Ang nag-bind sa histone ay si DNA. Okay? Konti na lang. Nucleolus. Okay? Parang ano rin tayo, face to face. Lagi yung naririnig yung konti na lang. Nucleolus generally is spherical, highly basophilic subdomain of nuclei in cells actively making protein. Kung sipag daw gumawa ng protein ng nucleolus. Intense basophilia, naintindihan na yan, is due to the presence of densely concentrated ribosomal rRNA that is transcribed, processed, and complex into ribosomal subunits in nucleoli. So, nucleolus. Okay, pero hindi po lahat ng cells visible ang nucleolus. Okay, hindi po lahat. Hindi nga lahat ng cells may nucleus. Okay, take note. We have here the three distinct components, the nucleolar organizer DNA, the pars fibrosa, and the pars granulosa. Okay? So, unahin po natin si nucleolar organizer DNA. Imaginein mo, may organizer daw. Okay? Is a pale staining region sequence of bases that codes for rRNA. Okay, nucleolar organizer DNA. The pars fibrosa are densely packed 5 to 10 nanometer ribonucleoprotein fibers composed of transcripts of rRNA genes. Pars granulosa consists of 15 to 20 nanometer granules. Granulosa kasi granules. Fibrosa kasi ribonucleoprotein fiber. Okay? Don't forget. Three distinct components, nuclear organizer DNA, pars fibrosa, and the pars granulosa. Okay? Wait lang po. Okay. So, cell cycle. Pag sinabi kong cell cycle, remember this is a repeated cycle, okay, of macromolecular synthesis. Repeated cycle. Kaya nga tinawag na cycle. Diba? So, guys, in cell cycle, tandaan po natin, this one facilitates in the growth and in cell division. Okay? So, bakit daw kailangan ng cell cycle? For growth and for cell division. Kasi guys, kung walang cell division, good luck ka naman, di ba? Importante ang cell division sa katawan natin. Okay? Paano kapag nasira yung isang cell? Wala na ba siyang replacement? Paano kung tumanda na siya? Di ba? Ma'am, meron naman pong autophagy. Guys, still, kailangan mo pa rin ng cell division. And remember, cell division is also important in reproduction. Okay? So, ma'am, ano po yung tatandaan ko? I want you to remember, we have 4 distinct phases. So, ano po yung 4 distinct phases? We have the G1, okay, the S, the G2, and the G0. Okay? Ano lang to, summary to. Okay? Ma'am, anong tatandaan ko dyan? Guys, I want you to remember, ito po yung nakikita natin sa mitosis. 4 distinct phases. G1, S, G2, G0. Ma'am, sino si G1? Alam mo ba that G1 is the longest? Okay? It is the longest and most variable phase. Okay? It is the longest and the most variable phase. Okay? Patandaan po natin yan, ha? Bakit mo kailangan ng G1? Remember that your G1 is important, okay? Because it permits repair. So for repairment of cells po pala si G1. Kaya kung depende kung gaano kasira, ganon din katagal gagawin. Kaya nga, it is the most variable. Okay? Take note. Next. S. So ma'am, ano ibig sabihin yan? Okay? Guys, do not forget, pag sinabi po natin S, it is for synthesis. Okay? For synthesis. Synthesis of what? Synthesis of DNA. Synthesis of DNA. So, tandaan po natin yan. Okay? Synthesis, o madaling tandaan, S kasi synthesis. Next, we have the G2. Sino po ba si G2? Okay? Alam niyo po ba, in G2, okay, ito po yung gap. Okay? Ito yung gap between DNA duplication. Okay? And the next mitosis. Gap between DNA duplication. Kasi guys, syempre ang sinundan niya is synthesis of DNA. It is the gap between DNA duplication and the next mitosis or next cell division. Okay? So, tandaan po natin. Next, G0. Alam niyo po ba, this is the suspension of cell cycle activities. Okay? This is the suspension of the cell cycle activities. guys, this suspension may be temporary, okay, or permanent. So ito ay pwedeng temporary, or permanent. Okay? Temporary or permanent yung suspension na yan, tandaan po ninyo. Okay? Kaya nga meron po tayong tinatawag na quiescent and senescent. Senescent. Anong ibig sabihin ng quiescent? Quiescent means reversible. Okay? Kunwari, enough na naman yung number of cells. Gagawa ka pa ba? Enough na nga. Okay? In senescent, this one is irreversible. In short, kulang na ng cell. Nagsimatayan na yung mga matatanda. Okay? And yet, kailangan na ng katawan mo na gumawa ng bagong cycle. Pero hindi na siya capable. That is senescent. Okay? Tandaan po natin yan. The difference between the quiescent and the senescent. Okay? Take note. Now, meron pa pong kulang dito. Ano yun, ma'am? May M. Ma'am, sabi mo 4 distinct phases eh. Okay? 4 distinct phases. Eh guys, tandaan natin, it is a cycle. Ha? Cycle po. Okay? So, meron po tayong M phase. So, meron tayong G1 phase, S phase, G2 phase, G0 phase, and then the M phase. Now, sino si M phase? Ito guys, very obvious. Ito yung sa mitosis. Okay? So, alam na natin yan. May G1, longest, di ba? And remember, most variable kasi depende kung gaano kasira yung cell. Ang cell, syempre, napapagod din yan. Okay? May nasisirang parts. Diba? So, kailangan ng repairment. Okay? So, G1. S for the synthesis of DNA, which is required in cell division. Okay? G2, gap between the DNA duplication and the next mitosis. G0. So, ito yung mismong suspension, kaya nga 0, di ba? So, it could be temporary or permanent. Kaya nga meron tayong quiescent and senescent. And then the last one is the M phase. Okay, the M phase. Yeah. Okay. Ma'am, paano naman po yan ngayon? So, kung titignan po natin sa cell cycle, okay, meron po tayong G1, okay, G1 tapos S phase, tapos G2, okay, so, meron po tayong G0, okay, then meron po tayong M phase, okay, so, ma'am, anong ibig sabihin yan? Okay? Ma'am, may nabasa po ako walang G0. Ito guys ay nakadepende kung anong kailangan ng katawan mo. It really depends based on how many cells must be synthesized. Okay? Ilang bagong cells ang kailangan natin. Okay? Ito ay nakadepende. Kaya nga minsan, after G2, M kagad. Wala nang suspension. Okay? Wala nang pagpigil ng paggawa ng bagong cell. Kasi depende yan kung gaanong renewal ng cells ang kailangan mo. Okay? And then guys, dahil medyo familiar na tayo dito, di ba? Dito na rin po papasok si cytokinesis. Okay? Saan? Yung cytokinesis bago po mag-G1. Okay? So remember guys, the cytokinesis, this is the division or cytoplasmic division nabanggit natin kanina. Okay? So, ibig sabihin, nag-undergo na siya ng cell division. Kaya meron ka ng cytokinesis. So, yung nag-undergo na cytokinesis, yun yung pupunta or babalik sa G1. Okay? So, next. Konti na lang to. Okay. So, mitosis. Anong ibig sabihin daw ng mitosis? Okay? Remember, pag sinabing mitosis, okay, you will have a two daughter cells. Okay? Two daughter cells. So, ano ibig sabihin? What is the difference between meiosis and mitosis? In mitosis, you have two daughter cells which are identical to the parent cell. Okay? Identical to the parent cell. Okay? Take note, mitosis. Okay? So, ma'am, ano pa po yung tatandaan ko dyan? Okay? Ano pa po tatandaan ko dyan? Remember guys, in mitosis, meron po tayong mga different phases. Okay? The PMAT, if you're familiar with PMAT, diba? The prophase, metaphase, anaphase, telophase. Okay? So, ma'am, para saan yung mga prophase, okay, metaphase, anaphase, okay, and then telophase. Okay? So, guys, i-highlight ko lang po sa inyo kung ano yung nangyayari sa kanila. Okay? So, simulan po natin kay prophase. In prophase, I want you to remember, if this is your cell, okay, you have the nucleus, the nucleolus, okay, and then the cytoplasm with the organelles. Okay? So, simulan natin kay nucleolus. Alam niyo po ba that during the prophase, okay, that during prophase, the nucleolus disappears. Nawawala daw si nucleolus sa mismong prophase. Okay? Yan ha, tandaan po natin yan mga kapatid. Yung mga highlights lang po tayo. Ma'am, ano pa po yung tatandaan ko? Now, let's say you have here the chromosome. Kasi, syempre, may DNA ang nucleus, di ba? Okay? So, nandito po ngayon si chromosome. Ito si chromosome. Okay? Chromosome. Okay? So, meron po tayong chromosomes dito. Okay? Ang tawag po dyan, di ba, sister chromatid. Okay? Kasi guys, ang isang chromosome, ganito itsura. Yan. Okay? Dalawa, kaya nga nag-X yan eh. Okay? Sister chromatid. Alam niyo po ba? Okay, ayan ha. Alam niyo po ba that your centromere, okay? O, di ba natatandaan niyo si centromere? Okay, yung may centriole, okay, centromere. Okay, nasaan daw si centromere dito? Andito sa part na to. Bakit? Kasi si centromere, siya po yung pinaka-constricted region. Constricted region of chromosome. Okay? Constricted region of chromosome. Okay? So, ma'am, ano po yung tatandaan ko dyan? Kasi guys, gusto ko maintindihan po ninyo na na paano ba nangyayari dyan sa bawat case na yan. Madaling magkabisa, I'm telling you guys. Pero gusto ko lang po na i-highlight yung mga certain parts na hindi natin na-appreciate talaga. Diba? Ano po yung gusto ninyong i-highlight pang isa? Alam mo ba, para magdikit yung dalawang chromosomes na yan, okay, bago sila maging isa, kasi sister chromatid ka nga, meron siyang kinetochore. Okay? So, ito po yung kinetochore. Kinetochore. Okay? Kinetochore. So, ma'am, sino yung kinetochore na yan? Alam mo po ba that kinetochore is the site of attachment, okay? Site of attachment to microtubules. Di ba natatandaan niyo si microtubules? Kani-kanina lang yun. Okay? So, guys, ibig sabihin, kung medyo iayos natin, parang ganyan daw ang itsura niyan. Di ba? So, nandito sa gitna na to, si kinetochore. Okay? And yung kinetochore na yun, sabi nga, it is the site of the attachment to microtubules. Okay? So, madali lang, di ba? As in, madali lang. Yung highlight lang to, mga kapatid. So, next. Ano pa po? Ay, number ang tutuloy. Next. So, alam mo na kagad na nawawala yung nucleolus. Okay? Alam mo na mayroon po tayong chromosome, di ba? Kung ano yung sister chromatid, sino si kinetochore. Okay? Guys, I want you to remember, okay, na dito nyo na po ma-observe sa mismong prophase. Okay? Di ba may ganyan? Yung centrioles natin, natatandaan ninyo kanina, hindi ako magaling mag-drawing. Okay? It will now have the mitotic spindle. Okay, magkakaroon na po ng mga mitotic spindle. Okay? So, yun pala yun, no? May mga mitotic spindle na tayo. Okay? Then, guys, do not forget, okay, na yung mga kinetochores natin, okay, na naka-attach po dun sa microtubules na naka-attach sa kinetochore. Okay? Ito ha, ulitin ko lang. Okay. So, natatandaan po si centromere, di ba? Si centromere, what else? Si kinetochore, okay? Si kinetochore, kung saan daw naka-attach si microtubules. So, ito si microtubules, ito si kinetochore. Kineto. Microtubule. Okay? So, para lang po maliwanag. So, ma'am, ibig sabihin, nagkakaroon na po ng formation ng mitotic spindle during the prophase. Yes. Okay? So, madali lang, di ba? Next, how about in meta? Okay? Metaphase. Sa metaphase, tandaan po natin, madaling tandaan. Bakit? Middle. Nasa gitna. Bakit? Kaya nga guys, ganito itsura lagi ng metaphase. Isa sa pinakamadaling ma-identify yan. Anong karakteristik ng metaphase? It is the presence of equatorial plate. Nasa gitna, middle. Okay? O, yun na yun. Di ba napakadaling tandaan? How about in the anaphase? Okay? In anaphase, guys, madaling tandaan. Lahat naman madaling tandaan, ma'am. Away. Di ba nasa equatorial plate na kanina? Ngayon, guys, maghihiwalay na tong sister chromatid natin. Okay? Maghihiwalay na siya. So, ibig sabihin, magiging ganito. Pahiwalay na. Okay? So, away. Kaya nga, away. Diba? Anaphase, away. Okay? And then the last one, telo. Okay? Anong meron kay telophase? Magkakaroon na ngayon ng nuclear envelope. Okay? Kasi kailangan maprotektahan yung nucleus. Kasi nandun yung DNA. Di ba? So magkakaroon na siya ng nuclear envelope. Remember, one of the characteristics of telophase is the presence of a cleavage furrow. Okay? Ito si cleavage furrow. Okay? So, guys, dito na lang sa isang side. Di po ba, meron ka na dito mga chromosomes with kinetochore. Ayan, nandito na siya. Okay? And then, magpo-form na siya ng nuclear envelope. Okay? So, yun na yun. So, mabubuo na siya. Tapos, magpe-pairing na yan, microtubules. Okay? Tapos nun, madidivide. Then, you have 2 daughter cells. That's it. Di ba? Parang, wow, ang dali lang pala, bilis lang pala yan. Ang dami-daming pinakakabisag before. Okay? Pero yun lang pala, ganun kasimple lang. Di ba? So, madali na sa inyo, most especially in metaphase and anaphase. Isa yan sa pinakamadaling i-identify. Okay? As in, isa yan sa pinakamadaling i-identify. Now, how about for meiosis? Okay? Meiosis tayo. So, in meiosis, guys, tandaan po natin, no? May difference po kasi si meiosis and mitosis. Okay? Hindi mo pwedeng sabihin na, ma'am, parehas lang naman yan. Guys, hindi po eh. Okay? Ma'am, bakit? Alam niyo ba kung kailan lang kailangan si meiosis? Okay? So, sige nga, kayo nga po, tanungin ko. Tuwing kailan nagkakaroon ng meiosis? Do you have any idea? Okay, kailan po nagkakaroon ng meiosis, mga kapatid? Okay, any idea? From MP21? Ayan. So, sabi, okay, very good, no? So, fertilization, sex cells, good. Okay, very good na, naging good pa. Very good. Okay, so ma'am, ano ibig sabihin? Ito ay very important. Okay, sa meeting, ayan na, sa pag-meeting, or sabihin na natin, pag-mating, meet, di ba? O mapahayop, mapatao, same. Di ba? May meiosis for reproduction. Okay, ma'am, paano nangyari yun? Anong difference niya kay mitosis? Guys, kasi tandaan natin, in mitosis, you have 2 identical daughter cells, which is identical to the parent cell. Okay? Identical to the parent cell. Okay? Ang meiosis, guys, tanungin ko kayo, kayo bang magkakapatid, magkakamuka? Di ba? Hindi. Okay? Ma'am, may kambal po sa amin, ibang usapan yun. Okay? Pero, dapat hindi talaga kayo magkakamuka. Okay? Hindi kayo magkakamuka. Meiosis, meron kang special characteristic, remember. Okay? meron kang special about you, maybe your eyes, your nose, okay? Your complexion, 'di ba? Maaaring kahit may kakambal ka, may iba sa inyo. Okay? Meiosis. Kahit ang nanay mo, okay? Sabihin, ma'am, para kayo may pinagbiyak na bunga ng nanay ko, still magkaiba pa rin kayo ng itsura. Okay? So, take note. So, dito po sa meiosis, nagkakaroon po tayo ng genetic variation. Okay? So, ma'am, question. Paano naman po yung meiosis na yan? Okay? Paano yung meiosis na yan? Guys, medyo parehas lang naman sa mitosis. Pero may i-highlight lang din po ako. Okay? So, simulan lang po natin sa simpleng addition. Okay? Ma'am, bakit? Remember, di ba? Lahat po tayo, okay, except for some with some genetic abnormalities, lahat po tayo may 46 chromosomes. Okay? Lahat po tayo may 46 chromosomes. Okay? Very simple. Okay? Kasi, guys, tandaan po natin, 23 comes from your father. Okay? And 23 chromosomes comes from your mother. Okay? Share ko lang to ha, as in share ko lang. Maisingit, para lang aware kayo, no? Kung mahilig po kayong manood ng mga Discovery Channel, mga ganyan, di ba? Feeling ko, baka napanood na ninyo. Okay. Paano ba yan? Ayan. Mayroong DNA, okay, or mayroong nakuhang blood na lang. May nakuhang blood dun sa area na yun. Okay? So, guys, ito sample lang ha. Sample lang. And sila nagsabi nito, hindi po ako. Sinasample ko lang para mag-gets lang po ninyo. Okay. And then, nung chinek nila, overall, meron po siyang 24 chromosomes. This is how I write chromosome. No? Shortcut yan. Shortcut ko yan. Okay? So, 24 chromosomes daw. So, syempre guys, alam niyo na kapag inad ninyo ang 23 and 23, ang makukuha ninyo ay 46. Di ba? Although meron tayong mga cases na may extrang X chromosome, kaya XXY, di ba? Or nawawala si X. Okay? So, parang ganon. So, may mga abnormalities. Pero eto guys, kakaiba eh, 24 chromosomes. Okay? Guys, alam niyo kung sino ang sinasabi nila or kino-conclude nila kung kaninong blood yun. Hulaan ninyo kung kanino. Baka mamaya napanood din yun. May idea kayo? Wala. Kayo nyo lang, baka alam niyo mga kapatid. 24 chromosomes. Pero tao ha? Tao. Okay, do you have any idea? Wala. Okay. So, guys, dahil baka hindi nyo talaga pa nakita yung ano nyo, they claimed or they actually concluded na si Jesus yon. Okay? Pero kung titignan kasi natin, di ba, 23 comes from the mother, which is tao naman talaga si Mary, di ba? Tapos, guys, missing halos yung sa father eh. Okay? As in missing yung sa father, except binigyan niya ng isang chromosome. Kasi guys, yung last chromosome, that is designed for your sexuality. Okay? That is designed for your sexuality. Kaya nga, yung 23rd chromosome na yun ang isa sa pinaka-importante para malaman kung ikaw ay babae o lalaki. Kasi sinabi na si Jesus daw yun kasi may +1 lang and yun yung nasa 23rd chromosome. O diba? So, meron kayong ma-impart sa mga kasama niyo dyan sa bahay. Okay? Aalamin ko kung saan ko yun napanood ulit para pakita ko talaga sa inyo. Although, meron naman kayo site dyan yata. Okay? So, yun. Di ba? Nakaka-amaze lang yung part na yan. Okay? Now, ma'am, gets ko na, ma'am, na 23 galing po sa tatay ko dahil sa sperm niya at 23 po galing po sa nanay ko dahil po sa egg cell niya. Okay? So, okay tayo dyan. Now, paano, ma'am, mangyayari merong 4 daughter cells? 4 daughter cells ang meiosis. 4 daughter. Because of what? TMAT. Ma'am, ang TMAT sa mitosis, ikaw na nga, ma'am, nagsabi, ano yun? 2 daughter cells lang. Tapos, ma'am, ang piniklaim mo ngayon sa meiosis, you have 4 daughter cells. Ayan. Ayun, meron daw sa YouTube. Ayan. So, check nyo na lang. Mahaba yun eh. Ilang oras din yata yun. Panoorin ninyo, maganda yun. Okay? So, 4 daughter cells. Okay? Sa meiosis, 4 daughter cells. Bakit? Because we have prophase 1 and 2, metaphase 1 and 2, anaphase 1 and 2, telophase 1 and 2. In short, 2 beses mo inulit si Pima. Ganun ang nangyayari sa meiosis. Okay? So guys, meiosis ang pinag-uusapan natin. Ma'am, paano po yan? Let's start with P1. When I say P1, this is your prophase 1. The characteristic of P1 is the crossing over. Okay? May crossing over na tinatawag. Ano yung crossing over? Okay? Crossing over. Guys, hindi ako magaling mag-drawing. Kayo nang bahala na umintindi. Okay. So, kasi given to, yung isa galing kay mami, di ba? Ito, kunwari, galing kay mami, galing kay daddy. Ayan. Okay? So, merong crossing over na tinatawag. Okay? Crossing over, ito po yung magde-determine ng different appearances ng magkakapatid. Yung crossing over. So ngayon, sabi mo na sa kapatid mo, kaya pala hindi tayo parehas matangos ilong, kaya pala pangu ka. Because of this prophase 1, the crossing over. Diba? Totoo yun, mga kapatid. Okay? Ibig sabihin, nag-cross over. Kunwari, ito yung isang kunwari, ganyan. Okay? Ayos natin. Kung maayos ko. Crossing over. May gulay. Ano kaya? Yan. Feeling ko makatotohanan naman. Yan. Yan. Feeling ko makatotohanan yung crossing over ko. Okay? So, ito yung, kunwari, ito yung kay mami. Tapos, nandito yung kay daddy. Ayun. Yun ang crossing over. O, diba? Yun ang ibig sabihin yan. Okay? Yun ang ibig sabihin po nitong crossing over nito sa prophase 1. Sa mitosis, walang 1 ha. Okay? So, ma'am, what's next? So, yan ang characteristic ng P1. M1 or metaphase 1, okay? Ito similar kay mitosis. Ibig sabihin, magkakaroon ng pairing, okay? So, ibig sabihin, ito, na-crossover. Sila na yung magpe-pair. Okay? Sila na yung magpe-pair. Okay? So, guys, take note, dahil nag-pair na sila, nag-pair na. Okay? So, parehas na siya sa mitosis. So, lagay ko lang dito, mitosis. Kung ano yung characteristic na binigay ko sa inyo, yun din ang mangyayari kay metaphase 1. Okay? Then, how about for A1? Okay, anaphase 1. Similar din, parehas din sa mitosis. Okay? And telophase 1, meron ka ng 2 daughter cells. Pwede ma'am 2 daughter cells? Hindi eh. 1 ka pa lang eh. May 2 pa. Diba? So ma'am, anong mangyayari sa 2? Okay, anong mangyayari sa 2? Simulan natin kay P2. Okay? Wala nang crossing over. Okay? Wala nang crossing over. Wala na nito. Bakit? Kasi nakuha mo na yung characteristic. Okay? Or yung chromosome na galing sa nanay or sa tatay mo na nag-mix. Diba? Hindi kapag ano pa yan, nag-crossing over pa yan, panibagong itsura na naman yun. Baka nasa loob ka pa lang, nagmo-morph ka na, diba? Kasi di ba ang crossbreed, hindi kayo same breed. Ang crossing over is kasi ang tanong, parehas lang po ba yung crossing over at crossbreed? Yung crossbreed, sa bagay, I could say na may similarities. Kasi ano eh, kasi wala namang parehas na tao. Lahat tayo may pagkakaiba. So, lalo na sa asawa, di ba? I mean, sa partner mo. Kumbaga, feeling ko nga, oo. Baka. Alamin ko yan. Pero, yun nga, walang tao na identical sa isa pang tao. Okay? So, yun. Baka nga. Okay? Kasi ginagamit lang natin yung term na, na crossbreed sa mga hayop eh. Okay? Baka. Okay? Alamin natin yan. So, in prophase 2, no crossing over. Okay? Tapos, yun lang na iba dun. Tapos, in M2, ganun ulit. No? In M2 or metaphase 2, same ulit, mitosis. Okay? Tapos, A2, ulit-ulit, mitosis. Tapos, sa T2, may 2 daughter cells ulit. Which is now, meron ka ng 4 cells. Kasi meron ka ng initial, meron pang sumunod. So, 4 cells. Okay? So, yun po yung ibig sabihin po nito. Okay? Tapos, meron pa yan meiosis 1. Pag sinabi mong meiosis 1, ito yun. Meiosis 2, ito yun. Okay? Hindi po kadalasan ginagamit ang prophase 1, prophase 2, or metaphase 1. Hindi. Madalas po siya meiosis 1 and meiosis 2. Kasi isang isang buo yun eh. Pwede mo bang sabihin na meiosis pero wala kang metaphase? So, that's impossible. Okay? And then, dito po nagkakaroon ng mga possible genetic disorders. Like yung mga Down syndrome. Diba? So, dito yun nangyayari sa meiosis mismo. Okay? Kaya nga, di ba, sa Down syndrome, yung trisomy 21. Okay? Ibig sabihin, may sumobra ka. Okay? Maraming mga genetic disorders eh. Yung mga Klinefelter, di ba? Klinefelter syndrome. Tingin ko sa cytogen, maaaral ninyo yun. Okay? Mga Turner syndrome. Okay? So marami po, hindi lang Down syndrome ang pwedeng pumasok po dito. Okay? Marami po yan. So sa cytogen niyo po yan. Okay? Para mas ma-explain sa inyo. Okay? So ito po yung pinaka summary ng cell cycle ng mismong cell division. Ayan, ito po yun. Okay, but I will provide